Up next, the evolving landscape of pulmonary arterial hypertension trials. Part 1, Progress in Trial Design, chaired by Marty Gomberg-Maitland, Washington, USA, and Marc Humbert, Paris, France.
Hello, this is Marc Humbert speaking from the Université Paris-Saclay French Referral Center for Pulmonary Hypertension. I'm delighted to co-chair this session with Dr. Marty Gomberg-Maitland from the George Washington University. Our topic today is very timely and exciting. We will discuss the evolving landscape of pulmonary arterial hypertension. In this first part, we will cover the progress in trial design, and we will start with a few presentations, followed by a multi-stakeholder debate with representatives from all horizons. So let's get started with the first presentation. Hi again. So I'm going to open this session with a brief introduction. I would like first to share my disclosures with you and briefly set the scene. I mean, we are all very knowledgeable of the pulmonary arterial hypertension pathophysiology and management. However, one has to bear in mind that we are dealing with a chronic condition characterized by pre-capillary pulmonary hypertension, and it is caused by a combination of many factors obstructing the small pulmonary arteries, including progressive structural remodeling of the small pulmonary arteries. This will lead to right heart failure, exercise limitation, and if left untreated, death. We know that this condition, PAH, is rare. The prevalence in Western countries ranges between 15 and 50 per million. The incidence is around 6 per million per year in the Western world. The pathophysiology is very complex, but there is a very clear indication that pulmonary artery endothelial cell dysfunction is of paramount importance in PAH. And thanks to the very nice understanding of this, and thanks to a very strong translational research, 14 agents have been approved in the last 25 years with an orphan drug status to treat this condition. That being said, for patients who remain refractory to medical therapy, lung or heart-lung transplantation is still available for some patients. The field of PAH has been a field of great success in terms of drug development. We know for sure that three major pathways are dysfunctional in PAH, namely the endothelin pathway, the nitric oxide pathway, and the prostacyclin pathway. And in recent years, it has been possible to develop several agents targeting one of these pathways or the other. And in the last 7 years, a very important advance in the field of PAH has been a better understanding on how to combine drug targeting different pathways together. We should acknowledge here the strong background vascular biology in the field of PAH, and there have been many Nobel Prize awardees working in the field of pulmonary vascular and systemic vascular biology. And many of these discoveries have allowed development of drugs interfering with one of the three pathways at play in PAH. Thanks to a very fast translation from basic science to pharma and to clinical trials, it has been possible to publish in major journals key articles which have led to the approval and reimbursement of different drugs. What is really interesting when one reads the literature in that field is that we have a lot of endpoints which have been used to convince the community of the meaningful results of the drug in the evolution of PAH. 6-minute walk distance has been by far the most widely used endpoint in the field of PAH, and it has led to quite a lot of approvals. Time to clinical worsening has been also used quite a lot since the early 2010s. And of course, survival has been studied in many studies, but this endpoint has never been, in fact, the primary endpoint of randomized controlled trial, but it has been reached in the very first published randomized controlled trial on prostacyclin in what was called at that time primary pulmonary hypertension. Despite drug discovery and development, PAH remains a very severe and devastating condition. So it really emphasizes the reason why we gather today in order to work together to improve phase 3 trials in order to offer the community more modern management for PAH. New developments have been done in recent years, and many companies try to develop new drugs or new dosages of drugs targeting the well-established 3 pathways. So either higher doses of some drugs or fixed combination of 2 approved drugs or inhalation of some drugs or variation of some drugs. So this is interesting. This is certainly the way to go to simplify, improve, and refine coverage of the 3 pathways. But clearly, we are plateauing, and we need to think outside the box and develop new targets. And there is no shortage of new targets. There are many basic labs and translational labs working on the pathological drivers of PAH. We know that dysregulation of vascular tone remains a very important abnormality to target, but abnormal proliferation and remodeling is certainly a current major ambition to target in our field. Reverse remodeling should be the way to go. We have a lot of drugs in development based on the better understanding of genetics, remodeling, inflammation, metabolic, humoral, and many other pathways disturbed in PAH. And clearly, all these novel treatment targets are very exciting. Not all of them will be successful, but all of them deserve a careful analysis on how to test these pathways, how to test these new compounds, and how to reach a meaningful conclusion on if and how we should use these new approaches in the future. So we have novel drug therapies on the horizon. We know that the major challenge remains to prioritize drug discovery and development. And this is the reason why we are together today. So this is my final slide. So the objectives of this meeting, and I thank you warmly, first for hosting us at CVCT and second for joining. We want to share the progresses in PH trial design, especially phase 3. We want to redefine clinically meaningful endpoints in PH. We want to discuss how to redesign PH trials, and we want to discuss what endpoints are clinically relevant regulatory approvable, and economically valuable. So the part one of the session will end up at 5:30 CET or 11:30 ET. It will be co-chaired by Marty and myself, and then the second part will be co-chaired by Dr. McLaughlin and Dr. Simoneau. So now it's my great pleasure to hand over to Marty Gomberg to introduce the next speaker. Thank you, Marty.
So I'd like to welcome Nazareno Gallier. He's from Bologna, Italy, and obviously well known in the field and been doing this for quite some time and always has some challenging questions and thoughts. His talk is going to be traditional design of phase 3 PAH therapies.
Thank you, Marty. Thank you, Mark, for the presentation. Let's go to discuss about the traditional design of phase 3 randomized control studies. So this is the time course of the 45 studies performed in PAH patients. And as has already been said, the majority of them had some primary endpoint, the 6-minute walking distance, and only 6 clinical worsening. And these are the studies with 6-minute walking test as primary endpoint. As you can see, were quite short study, up to 20 weeks, and small sample size. Maximum was 405. In 2008, at the Fourth World Symposium on Pulmonary Hypertension, there was some recommendation for new design, and it was suggested to utilize clinical outcome as primary endpoint. And this is time to clinical worsening is a composite endpoint, is the time to the first event and events where mortality, non-elective hospitalization, and disease progression. This led to longer and larger studies with an elevated number of clinical events and possibility to make also analysis of different components and also a meaningful subgroup analysis. And as you can see on the right, in orange, the studies who have been performed with outcome as an endpoint. And as you can see, the sample size is larger, but not so larger as expected. For example, some of these studies had around 406 or 700 patients as a global sample size. And just to show you the time to clinical worsening of the Seraphine study with macitentan and the Griffon study with selexipag and the Ambition study with head-to-head comparison of initial combination therapy with initial monotherapy. And this is the last study published, the Freedom EV study with oral treprostinil. Now, at the 6th World Symposium, there were some discussion about clinical trial design and new analysis of these studies were proposed according to the phase of the study. So for phase 1 and 2 was suggested to include the pharmacokinetic and pharmacodynamic studies, so some biologic and mechanistic markers. Also, the trial should use risk tools to phenotyping better and reducing the heterogeneity of the included patients. And also novel efficacy markers in phase 2 were suggested as secondary endpoint like magnetic resonance imaging and other imaging parameters. For the phase 3 study, it was suggested to include not only time to clinical worsening, but to change in time to clinical improvement, which usually utilize the same parameters, but in the opposite way. And also there was a suggestion to utilize again risk as a marker of efficacy in that phase as an exploratory endpoint. And this is what I will explore more in depth because it's for me a very interesting suggestion. So we utilize the stratification which identifies the risk, so the annual mortality of the patients in low risk and intermediate risk and high risk. This is the table of the current guidelines of the European Society of Cardiology and European Respiratory Society. There are different possibilities to utilize this table, providing meaningful results. And now when we analyze endpoint for clinical trials, we need to remember some terminology like biomarkers, which are measurements of biological processes, correlate, which is a biomarker which show statistical correlation with a true clinical efficacy measure like mortality, like hospitalization, and surrogate. Surrogate is a biomarker used as a substitute for a true clinically efficacy measure. And risk stratification is surrogate endpoint. This is what we can ask to us. So risk stratification is correlated in retrospective registries analysis with mortality, but no surrogate endpoints for PAH randomized controlled trials have been validated, including 6-minute test, hemodynamics, and NT-proBNP, which are largely utilized in the stratification of the risk. So again, surrogate endpoint is a parameter which should intercept the effect of the intervention leading directly to the true clinical endpoint. However, we know that some intervention may have unintended negative effect to the true clinical endpoint, or may have alternative beneficial effect which is not captured by surrogate endpoint. So it's very important to realize that a correlation does not surrogation make. This is an example, the IMPRES study with imatinib, with increased exercise capacity and improves hemodynamics, reducing pulmonary arterial pressure, increase cardiac output, and reduce pulmonary vascular resistance. However, despite this important effect, the outcome is not improved and is worse as compared to placebo. So again, a measurement of the surrogate is a measurement of clinical sign used as a substitute for a true clinically efficacy measure. In order to prove that the correlate is a surrogate, we need to prove that it fully mediates the effect of the intervention on the valid clinical endpoint. And the Prentice criterion says that essentially this parameter it requires the surrogate variable to capture any causal relationship between the treatment and the true clinical endpoint. So surrogacy is very difficult, unfortunately, to demonstrate. This is why experts and regulators utilize a sort of hierarchy for evidence. So if you utilize correlate, this is the level 4, the lowest level of evidence for effect. Then there is we can utilize non-validated surrogate, but also this is not good level of evidence. If we utilize validate surrogate when we found one, like blood pressure for drugs, for antihypertensive drug, we should we can avoid to utilize the true clinical events because it has been demonstrated that blood pressure is a surrogate. At the end of the scale of evidence is the utilization of true clinical measure like death, like hospitalization, and other important clinical parameters. So the analysis of the use of outcome as primary endpoint in pulmonary arterial hypertension require direct measurement of the clinical component, either time to clinical worsening or time to clinical improvement. But only this measurement, direct measurement, is give us the possibility to utilize this parameter. So clinical components should be prospectively defined and blindly adjudicated. And no surrogate endpoint for PAH RCTs have been validated, including 6-unit walking distance, hemodynamics, NT-proBNP alone or combined in risk stratification tools. So a correlate does not, again, surrogate. Thank you.
Thank you very much, Nazareno, for presenting very nicely the traditional approach. And now we have a few hours to discuss how we can improve that. So it's my great pleasure to introduce my co-chair, Dr. Marty Gomberg-Maitland, who is going to discuss target patient populations, phenotyping and enrichment aimed at maximizing response rate. So thank you, Marty.
Thank you, Mark, for the introduction. And as always, it's a pleasure, of course, to host with Mark. So I thought I'd start by thinking about what this title means, because it's kind of a mouthful. And what I thought was, it doesn't matter what cohort we're doing a clinical trial in, but especially in rare disease, I think it's really important to understand the similarities and the differences between our patients to highlight who will benefit most from treatment. And I think that when we started trial design, we had nothing, and now we really need to target our therapies to design efficient trials, to improve clinical outcomes, and to target therapeutics. So why now? Truth be told, this is not a new concept. This is a slide that I presented at the CHEST meeting in 2009, and it was on repurposing drugs and trial design. And you can see that many of the problems that we're going to discuss today and the reasons for why we need to enrich existed back in 2009. But today, there's a difference. And that's because we moved from short 4-month trials without making it an event-driven trial to event-driven trials. We now have regulatory support to encourage the use of prognostic enrichment for phase 3 to improve the odds of success, limit time to enrollment, and limit time on placebo. It's still a rare disease with limited patient numbers. We have more therapeutic options, so it's really, patients are higher functioning and there's a higher bar to show benefit to get to phase 3, and we definitely need shorter trials, faster approvals, and less cost. So we've been using enrichment strategies. However, the majority of our strategies have really focused on disease severity stratification at inclusion. So we all know functional class, 6-minute walk distance, maybe subtype of PAH with connective tissue disease having more events, number of therapies and the types, class of therapy. And in heart failure, we use recent hospitalization as a method for risk stratification. In the remainder of this talk, which is, obviously going to be a bit short, I thought I'd highlight 3 topics. 1, adaptive enrichment. 2, use of composite risk scores. And 3, randomized discontinuation withdrawal studies. So what is an adaptive design? And I think most broadly, it's defined as any study that includes a design where a critical aspect of the study is altered based on the results, which implies that there will be an unblinding at some point at the trial, which means that you're going to have some kind of interim analyses. And the things that we can enrich for would be the number of patients, the study duration, endpoint selection, patient population, number of treatments, so that maybe you want to drop an arm or add a new therapy sometime during the trial. There can be a number of interim analyses. It just doesn't have to be one. And we can actually adapt the hypotheses based on the results. This is a typical adaptive enrichment strategy. You have the full population at stage 1. You do an interim analysis. You see which groups have the greatest benefit, whether it's a new biomarker or just a response to known endpoints, and then you enrich the population. So the first interesting study design that I thought we should talk about is an adaptive enrichment with continued enrollment. So what does that mean? It means that during this interim decision, we actually continue enrollment. We don't halt enrollment while we're analyzing the data, and we continue to enroll the patients that have the biomarker that we've selected. And this allows patients to continue enrollment and to enroll more patients with the biomarker that we think is going to have the greatest effect to measure the efficacy of the drug in the population. So it really, it's a method to save time and to increase the number of patients that we think will respond. Adaptive enrichment isn't just a form of adapting the design, but it's actually a strategy. So we've discussed now the enrollment strategy, which is you want more high-risk patients to have higher event rates, but you can also have a futility strategy, which is an analysis of the outcome. Maybe it's to show you that it's completely an ineffective drug. Maybe it's to drop a dose, or it's to alter the sample size. So next are risk scores. So this is a slide that I presented at the World Symposium on trial designs and new therapies for pulmonary arterial hypertension. And you can see it's the full and then the enriched population, but it's using risk scores that we validated in our retrospective data to actually stratify patients into low, intermediate, and high risk. So that's a concept, but how do we prove that that concept actually makes sense and that we should start utilizing it? And this was a great paper that just came out in the Blue Journal, and it used the French, the 3 risk scores, French, Compare, and Reveal, in 3 pivotal event trials, Ambition, Seraphim, and Griffin, and aimed to prove that risk scores can be used as a method for enrichment. And frankly, the statistical analysis is quite complex and sophisticated. And this is just really a brief summary of it, which is they used ROC curves to look at the performance of predicting clinical worsening within the pooled placebo cohort. They did power simulations to look at sample size and treatment time reductions. And they did a cost analysis. So this slide next shows the 3 scores, blue French, green Compara, orange reveal. And what you can see is 3 different cohorts. One was a mix of intermediate and high risk. One was half high risk and half everyone else. And then the third was all high risk. And what you can see is that whether it was a sample size reduction, the percentage, or the average time on treatment, so the length of the trial, clearly the high risk did much better than the other 2 cohorts with smaller numbers needed and a shorter trial. As far as cost effectiveness, they looked at the ratio of screening costs to research costs to see the potential net savings. And what they found was actually that the reveal, because they separated the different risk scores, intermediate and high risk had a 40% reduction in total savings, and it was a 1-to-2 ratio of savings. And then finally, the randomized discontinuation trials. This is one of my favorites, and I think it really does have a role now in clinical trial design. So what you can see here is there's a population, you have a run-in open label phase, and this allows us to determine if patients can even tolerate the drug. It also identifies super responders to the drug, and they'll continue on open label, and then you randomize the rest of the cohort to placebo or drug. And if the person progresses, then you can unblind and give them the drug if they were on placebo. The problem is that the assumptions that need to be met are you can't have a curative drug in the open label. You need to have reliable endpoints and surrogates in order to truly detect a real response. And it needs to be safe to withdraw medication after the open label and return to your previous clinical condition. And I think this is what's caused a lot of hesitancy in pulmonary hypertension drug development, because we are always worried about withdrawing. But you know what? Now patients are on 2 to 3 drugs always now as standard of care. And I think it'll be okay to withdraw during this kind of test phase 1, phase 2 period to see if you can even tolerate the drug. And I think we can use risk scores as a biomarker for the enrollment stratification. So I think enrichment strategies should be utilized now. I think we're starting to see that in some of our new phase 2 trials that are being initiated. I think it's really important that we have a reliable responder and high risk biomarkers to best stratify patients. And I think there's a way for us, like there's clearly room in the field to improve on that. I don't think we have the best biomarkers. I think we need to focus on that. I think that we can improve efficiency by doing this, and hopefully we'll see a new era of drug development in pulmonary vascular disease. I thank you for your attention.
Thank you very much, Mardi, for a very nice presentation again. The next speakers after the interim spot will be Dr. Paul Hassoun from Johns Hopkins University, and he will discuss seamless phase 2-3 trials. May adaptive design solve the problem?
Good morning. I hope you can hear me. This is Paul Hassoun. I'd like to thank the organizers and FAES for inviting me to this very interesting talk. I'd like to thank our co-chairs, Marty Gomberg and Marc Ambert. And can I have the next slide, please? Okay, these are my disclosures. The only thing relevant here is my position as scientific advisor to Merck, because I will mention a trial. I'm going to limit my talk to some definitions and look at pitfalls and advantages of adaptive designs. Next slide, please. So I'm glad that Marty Gomberg talked about adaptive designs. These are alternatives to traditional randomized controlled trial where the allocation of control and intervention groups are done according to a consistent randomization scheme throughout the trial. An adaptive seamless design allows modification to the processes and statistical procedures, usually at set intervals. And the goals obviously are to increase efficiency and lowering the cost, identify most promising treatments with the hope that we can save both time and cost. Next click, please. So seamless design typically combines two separate phase 2 and 3 trials. These are called operationally seamless. Adaptive seamless design makes use of data from patients enrolled in the phase 2 and before and after adaptation, and they can call inferentially seamless designs. These are based on Bayesian statistics using data from phase 2 to inform phase 3. Next slide, please. So cost and time reduction. Adaptive designs may help reduce the number of patients needed in phase 3, and I would show you that this is not always the case. Next click, please. It improves selection of target doses and participants in phase 3 trials. Next, please. And helps identify covariates between short-term endpoints, which are obtained in phase 2, and long-term clinical outcomes in phase 3. And next slide, please. With this process, you continue to follow patients on terminated treatments groups from phase 2 and throughout phase 3. And therefore, you can assess effects and safety, and change in treatment can happen where you can increase, and this will increase the safety and efficacy of the trial. Next, please. Adaptive seamless design trials have, this is really important, predefined interim points and predetermined modifications to study design based on observations during the trial. Next click. But there are potential problems. You can falsely detect treatment effects. You can, conversely, you can prematurely drop effective treatments, causing statistical or operation biases. And it is really important to emphasize that although you have predefined interim points and predetermined modifications, it is important that the statistical analysis at the predefined point is done in a blinded fashion. And there is a need for a data monitoring board. Next slide, please. So there are several adaptive design. Marty mentioned some enrichment based on phenotyping. You can have adaptive dose finding where in phase 2 trials, you have several doses, and that allows you to determine the most effective dosage. Adaptive hypothesis, where you can go from a single hypothesis to generating multiple hypotheses that you can test later in the trial or in phase 3 trial. Adaptive group sequential, where you can change the size of the cohort. You can make modification or deletion in the treatment arms, or you changing the endpoints. Adaptive randomization, the randomization scheme can be adjusted based on the results at the predetermined point to improve, to have a higher probability of randomizing individuals in the appropriate arm. I'll talk about seamless phase 2 to 3 design, where you go seamlessly from a phase 2 to a phase 3 trial. There are trials where you use biomarker adaptive trials, where specific biomarkers help identify patients that perhaps may respond better to a certain treatment strategy. Next slide, please. This is a very interesting review, a meta-analysis looking at over 140 trials. In the blue bars, you can see the seamless phase 2 to 3 trials, and they make up about 57% of these reviewed trials. Adaptive group sequential trials make up 21% of these trials. Biomarker adaptive, about 20%. You can see that although adaptive design trials have been used for several decades, there is an increased use of these trials in the past decade or so. Most or a quarter of these trials have been targeted to rare diseases where mortality is a common endpoint. And as mentioned by Marc Humbert previously, that's not a common endpoint in our PAH trials. Most of these trials have tested drugs, 85%, but also vaccines are tested, or medical devices. And I'm sure in the COVID development of vaccines, adaptive trials regarding doses or target populations have been used. Next slide, please. And this is from the same study. Looking on the left panel, you have the duration of the trial in weeks for phase 2 on the left, phase 3 on the left. It may appear that you can save weeks in terms of going from standard trial to adaptive trial. And you can also save or limit the cohort of individuals enrolled in your trial. But looking at all these trials in combination, there was not much saving in terms of duration of weeks and in terms of subject number. Next, please. Next slide. So moving on to an example of an adaptive trial in pulmonary arterial hypertension. These slides were given to me by Dr. Mahesh Patel. And this is an ongoing study using a soluble guanylate cyclase stimulator, same class as riociguat. But this is given by inhalation, which has a major advantage of acting locally in the lung and having very little systemic effects. This allows high target doses, optimizing efficacy. And this trial is currently in phase 2 for PAH, phase 2, phase 3, and also in phase 2 for pulmonary hypertension related to COPD. Next slide, please. So this is an example of an operationally seamless study to evaluate this compound, inhaled guanylate cyclase stimulator. And phase 2 allows for dose selection, and phase 3 for a confirmatory cohort. I won't go into the study population, but in this case, this is the typical PAH group 1.1 to 1.4, WHO functional class 2, 3, and 4, the typical hemodynamics inclusion criteria, and the 6-minute walk distance that is allowed between 150 to 450 meters. This particular trial had the advantage of enrolling patients on stable concomitant PH-specific therapy like endothelin receptor antagonist, phosphodiesterase inhibitor, or IV or sub-Q prostacyclin. Next slide, please. And so the difference between phase 2 and phase 3, as you can see, in phase 2, there are 3 doses of this inhaler tested versus placebo. So you have 4 arms. And the idea is to allow identification of a best dose effect for phase 3 in the confirmatory cohort. You can see that also the endpoints are changed from a phase 2 where the endpoint is pulmonary vascular resistance to a 6-minute walk distance in the phase 3 trial. And this trial may also allow identification of specific biomarkers as exploratory endpoints, like the NT-proBNP or a patient from WHO functional class. Next slide, please. I think this is the end of my talk. And again, I appreciate the opportunity to share my thoughts about adaptive trials. And I thank Marty for covering some of it in a very eloquent fashion. Thank you. Thank you, Mark. Thank you, Marty.
Thank you very much, Paul. We have now the next speaker preparing. So it will be Dr. Eric Vico from Université de Paris. The topic will be May Bayesian approaches help. So let's move on.
Hello. I first wanted to thank the FIAS and the chairman and co-chairman, and I will speak about the Bayesian approaches. So, okay, I go maybe, okay. Bayesian analysis present two different innovative aspects. I can say that there is something that is more philosophical regarding the change for advancement of medicine and what is a way to let the medicine advance or make progresses. And the other aspects are more technical and can apply to make easier different adaptive design, for instance. Well, if I go first to the philosophical aspects, the classical way to think about statistics, that is the frequentist statistical approach, is based on the fact that we think that medicine can advance by rejecting null hypotheses. For instance, the null hypothesis that the new treatment is equal to the old treatment, and we make a trial. And if we can reject this hypothesis in favor of the new treatment, we will change our practice. And it's very easy to conclude. This is a black and white conclusion based on a p-value. The usual p-value is 5%. The reason for the 5% is certainly not a scientific reason. It's really something that is more pragmatic or regulatory, but there is no, I would say, deep reason to choose this threshold value. Okay. The Bayesian approach is, I would say, philosophically completely different. We will have to estimate the probability of the unknown treatment effect based on the data. So it's not a question to reject one hypothesis. It's a question to estimate what can be the difference between two treatments. And then, based on this probability that I will estimate by a Bayesian trial, I will make my decision in order to change or not to change my practice. So it's not so easy because it's not black and white. It's something based on some kind of subjectivity. And obviously, this is various questions about the probability threshold that will make me change my practice, for instance. But what is important, I think, is that Bayesian, I would say Bayesian statistical analysis is strongly related to the question of the decision theory and not something that is just go by rejecting or non-rejecting hypotheses. If we see what are the combination between philosophical or technical aspects, there are some. First, Bayesian approach will incorporate what is known or believed before the trial about the treatment effect. When you are making a trial, it's very rare that you don't have any previous experience And what allows the Bayesian techniques is to incorporate properly on a formal, proper aspects this previous experience or previous belief. The other aspect is that it allows you to update information about the treatment when you incorporate new data. So that means that with Bayesian analysis, you can estimate the probability of the difference between new treatments. Well, we can say for each new patient that you have treated. And then combining what you knew before the trial and what is the data that you have accumulated during your trial, you have to the possibility to know what is the probability of the unknown treatment effect, and will support your decision with this probability. Well, the basis that we know something before, we will want to incorporate this in our analysis. We will combine the data known before with the data collected during the study to make the estimate of the probability that we want to use to make your decision. One critic, and very important critic, is that Bayesian analyses are too subjective because they depend on your opinion before the study. So we can consider its critics, but well, we can also answer that it's very important in medicine to consider also what was the previous information before your trial. And the other aspect is more technical, but you can consider to incorporate in your Bayesian analysis something that is absolutely neutral regarding your information before. So it's not mandatory to incorporate something that is subjective. You can also incorporate something that is very neutral. I will consider two examples regarding the comparison between frequentist and Bayesian analysis in the field of pneumology. Here, this is the OSCAR study that compare high frequency oscillation in IDS, and that's compared with conventional positive pressure ventilation as control. So the primary outcome was mortality at 30 days. The planned sample size was more than 1,000 patients. And the question is that since there are some problem of recruitments, OSCAR randomized only, well, almost 800 patients, and did not find any evidence of superiority of the experimental arm regarding the control arm. If we consider the same trial using a Bayesian analysis, and it has been made by Ryan and his team, what we can see is that using a Bayesian method, we will have get the same conclusion, but we would have saved more than 100 patients in comparison of what has recruited OSCAR, and we will have saved more than 40 weeks of inclusion to obtain the same conclusion. I will show you another example of comparison. This is the Eolia trial by Alain Combe for ECMO in severe RDS. And the conclusion of this trial was that ECMO was not superior to the use of the control treatment in these patients. And here, this is a post-hoc Bayesian analysis of the same trial by Gollinger and its team. And what Gollinger has shown is that we can calculate that the probability that the ECMO is better than the control group for mortality is 96%. So that means that, okay, it's not demonstrated on a frequentist approach, but you can estimate this probability as a very high value. This is the same if you consider an absolute risk reduction of 2% was 92%. So the conclusion is that ECMO has probably some benefit in this context. So the conclusion is in this way is rather opposite in terms of decision. So my conclusion is very simple, is that Bayesian analysis can help in order to obtain better conclusion for decision and more rapid conclusion for decision. Thank you very much for your attention.
Thank you, Eric. I think we all need to start using Bayesian approach. It seems that we're a little behind. With that, I welcome Steve Kaywood from University of Pennsylvania in Philadelphia. He's going to discuss single dose versus strategies comparison.
Thank you very much for inviting me to speak at this very interesting session and conference. I've been assigned multiple topics, and I'll try to be more provocative and leave time for others to speak rather than for me to speak. There's my disclosure slide. I've received funding from United Therapeutics to serve on a DSMB, and I've received funding for consulting from Vivus and Acceleron. The overarching theme, I think, to my talk as well as the other talks, is that the efficiency of randomized clinical trials in general needs to be acceptable for them to be ethical. And the efficiency can be considered a ratio of the research value of the obtainable data. This includes both the internal and external validity of the data, as well as the health impact, as a ratio to the costs of obtaining the data. And those costs include the risks and burdens on participants, the number of participants needed, and of course, the actual costs, financial and otherwise, in this time of scarce resources. So currently, the standard of care for treatment of pulmonary arterial hypertension is using effective drugs, at least in some countries. And I think it's inarguable, but we could discuss, that patients in a clinical trial should and need to receive the standard of care treatment as background therapy in order for these trials to be ethically justifiable. The other special circumstance might be using an active control. I think this generates several issues. Background therapy in most human disease makes it more challenging to find efficacy because you're usually looking at a smaller effect size. If you would think about doing a randomized clinical trial of insulin in a patient with diabetes who's untreated, you would expect a very large effect estimate conferred by insulin. On the other hand, if you were performing a clinical trial of a new diabetic therapy in patients already treated with insulin, you can expect a much smaller effect estimate requiring many more patients. Some regions do not have available or affordable therapy for pulmonary hypertension. And this brings up a lot of ethical issues, but namely, is it ethical to perform clinical trials without background therapy in a region where people are disenfranchised and it's not available to them? And this is a story that therapy for HIV and AIDS had to go through, and I think really serves maybe as a good track record for how we should be conducting studies in such countries. And then finally, standardization of background therapy will make our trial neater, increase the internal validity of the trial, but might decrease the external validity in that not all patients, when the new drug is approved, are going to be using exactly the same background therapy that was prescribed in the phase 3 clinical trial. Placebo versus active control is another interesting topic in pulmonary hypertension. So of course, randomization with placebo control provides the potential to estimate the effectiveness of a new or an unproven treatment or approach. On the other hand, using an active control compares two different treatment approaches, which are both thought to have an effect. And there are several flavors of using active controls, or several designs. One is a superiority design, where you're interested in comparing two drugs, which are both considered standard improvement treatments for pulmonary arterial hypertension, to see if one is better than the other. That's relatively straightforward. A little less straightforward is a non-inferiority design, where you'd like to study whether an alternative drug is not worse than the standard proven therapy in terms of clinical effect. The new drug of interest may be cheaper, easier to administer, it may have less side effects, so there may be some advantage to the new drug if you can show that it has the same effect as the old drug. And then finally, there is equivalency designs, where you're interested in drugs which are neither inferior nor superior, but really the same, and that's really less used. Well, there's lots of issues to discuss in this type of trial. Superiority trials will require larger sample sizes because both arms are receiving what's thought to be active treatment. Non-inferiority studies require very high power and higher precision estimates because you need to make sure that the confidence intervals of your new drug fall within some distance of the effect of the old drug. In addition, you need to have assay sensitivity. You really have to know that the active control, that standard drug, is effective. And if you were to include a placebo arm, the active drug, the active control would certainly be better. You also have the constancy assumption. This is a term which refers to the fact that the design of the non-inferiority study has to be identical to the study which showed the active control or standard therapy was effective. So you need to actually reproduce the same study that was used to prove that the active control worked. There's real importance to how you conduct these kinds of trials in terms of completeness of data and endpoint detection. Missing data or missing events could actually bias to the null and actually make the drugs artificially look similar, which actually in this instance is biasing away from the null. So the usual intent to treat approach may actually not be conservative, but maybe anti-conservative because it makes the arms look more similar. But of course, using a per protocol approach also has its own biases. We probably need to analyze these studies using both approaches. And finally, you don't want to perform an underpowered superiority study. And when the drugs look similar, call equivalency or non-inferiority. You really need to prospectively plan for this non-inferiority aim. Add-on versus replacement. So the traditional approach in pulmonary arterial hypertension has been to add therapies based on persistent symptoms or perceived risk factors for adverse outcomes. The REPLACE study was a recent open-label randomized clinical trial of changing from a PDE5 inhibitor to riociguat versus remaining on PDE5 inhibitors alone, which showed some benefit to the switch. This replacement strategy could decrease patient burden, could decrease side effects, and of course, could decrease costs of treatment. On the other hand, the add-on approach retains any possible benefit that you might be getting from the original therapy with the additional and potentially synergistic effect of the newly added therapy. Is it feasible to perform randomized clinical trials of add-on versus replacement strategies? Would the community tolerate this? And would investigators think this is worthwhile? And are these studies sufficiently justified? And finally, we're used to looking at single drugs in clinical trials, but what about strategies? and this was brought up in some of the previous talk, goal-directed therapy has been proposed to be an effective approach to treating pulmonary hypertension. But to be frank, there's really no data available to answer or support the fact that a goal-directed treatment approach is actually better than our standard approach to treatment of these patients. And in fact, when you look at other much more common conditions such as congestive heart failure or HFpEF, there really aren't great data supporting goal-directed approaches in those much more common conditions where there actually have been randomized clinical trials of this approach. So again, issues to discuss. Could we do a randomized clinical trial of goal-directed therapy versus usual care? What would be the best design for such a study, including what would be the goals? Would they be patient-reported outcomes? Would they be purported surrogate endpoints? Professor Gallier pointed out the issues with that. Would they be risk prediction rules? And could we come to an agreement of what would be the goal for such a study? And then there's these advantages and disadvantages of mandating a treatment approach. So being explicit about exactly which drug would be your first drug, which drug would be the second drug, and which drug would be the third drug, either drug or drug class, versus allowing more heterogeneity, supplying the goal back to the clinician and allowing them to decide what drug would be the next drug. Again, things to talk about. So thanks again for the time, and I appreciate your attendance.
Thank you, Steve. It was a great talk. Covered a lot in a short time period for sure, and lots of points of discussion for the panel. Next, I introduce Greg Elliott. He's in Utah, Salt Lake City, and he's going to talk about pragmatic post-approval trials and real-world data and how can we use this in pulmonary vascular disease.
Good morning here, and perhaps good afternoon or even good evening around the world. I want to thank Marty and Mark for inviting me to discuss pragmatic post-approval trials and real-world data. My goal this morning is to lay out for you the foundation and really to make a point that pragmatic post-approval trials and real-world data are useful throughout medicine. I'm going to focus a little bit on pulmonary arterial hypertension, but I'm also going to try to take a very broad view as I introduce this topic. First of all, I just want to touch on the background. For most of our newly approved treatments, the evidence from well-designed randomized clinical trials is not sufficient completely to guide physicians' choices of treatment for the patients that they confront every day. There are so many reasons for this. The patients often, of course, are much more diverse than the patients who are enrolled in the clinical trials. They may have multiple comorbidities that weren't well represented in the smaller sample sizes of the clinical trials. And we always wonder whether one of the patient's characteristics makes them different in some very important way from the patients reported in the clinical trials. Pragmatic trials and real-world data can provide evidence on the effectiveness and safety of treatments in clinical practice. And one of the things that we in the pulmonary hypertension community have to do, I think, is begin to ask ourselves, where are these important gaps in our knowledge and understanding of treatment? And this often comes, I think, from looking carefully at patients that are treated, large populations of patients that are treated for PAH, and seeing where there's wide variation in practice, which often will help us to identify some of those gaps in knowledge and understanding where clinicians are more or less left on their own to make decisions that they find reasonable and in the best interest of their patients. There are some essentials to pragmatic trials. First, every pragmatic trial has to have a careful thought about the setting. Steve's talk before mine was a perfect segue to my talk because he mentioned, just as one example, there are settings where patients don't have access to background therapies. They're settings where technologies are just not simply available, unlike tertiary academic medical centers. And yet these are very important settings for the treatment of pulmonary arterial hypertension. By the same token, the study population has to be a relevant study population. And we have already talked about phenotyping and characterizing the study population carefully so that we know who's in these trials. And then for the large pragmatic trials, we can begin to do subgroup analyses when we have large populations enrolled in pragmatic trials to look at subgroups, whether they be phenotype specific subgroups or other demographic characteristics that are subgroups, like elderly patients, or we've lacked good data for children for many years. The mode of intervention is very important. It depends a lot on what we're testing. It's maybe fairly straightforward to test medications, but let's just take the example that was shown earlier of meter dose inhalers. We have to be sure that in these pragmatic trials, there's proper training of individuals to use meter dose inhalers. Not all patients, have the same dexterity or training, and that's important. The comparator population has to be relevant. So, we want to pick a control population that is well represented of the disease and is receiving high quality care. And then ultimately, there's going to be discussion about the outcomes that we measure. And we've heard a lot already this morning or this evening about outcomes, but important outcomes also are sometimes patient-centric and relate to issues of quality of life, as well as quantity of life. We have to monitor the trial and we have to collect the data without changing usual care. And that's not easy in practice environments, but it's critically important that be thoughtfully designed with a pragmatic trial so that there's a minimum of interruption in the usual care that's delivered in practices that are built off of efficiency and the demand that all patients receive care through criteria of equity and diversity in communities. Next slide, please. Oh, I can do it. Thanks. There are many challenges to pragmatic trials. I've only listed two here. I'll mention a couple of others. But first of all, usual care may not reflect the standard of care. And that's an important issue to consider when designing a pragmatic trial. You want to be sure that you are achieving a high standard of care for patients with pulmonary arterial hypertension. You also, at the same time, want to be sure that you don't disrupt usual care so that what you determine in your clinical trial can't be reproduced after the trial. And monitoring and reporting safety events, which is required by regulatory bodies, can alter usual care just by the demands and the focus and the attention paid, as well as the time necessary. So these challenges and some other barriers, and people have listed barriers. They include barriers like informed consent, detailed informed consent. They've included barriers like lack of incentives for individual physicians in practice to conduct these clinical trials. They've included barriers like the inefficiencies that go on with clinical trials. All of these things have to be managed thoughtfully as part of a pragmatic trial so that the trial yields the answers that we want. And often what this means is that special designs are used, like clusters where medical centers are randomized rather than individual patients. There are some solutions here. And among those solutions will be creating research centers that are well organized to do this. I want to move on now to the question of whether electronic health records could provide real world data. And at least in the United States, Carrie Gilmire in Boston has taken us, I think, a step forward, first by showing that research efforts that rely on administrative data and were very limited by current coding systems that didn't adequately reflect pulmonary hypertension clinical classification scheme that we all use, and algorithms to identify PAH in administrative databases were widely variable, and very few of the ones that she studied were even validated. So her final conclusion of her first study was that ICD codes performed poorly, potentially leading to biased results because they weren't really capturing the population of interest. She's conducted a follow-up study, and the important finding, I think you can too, really on the top line, you can see the very low positive predictive value for the ICD-9 codes. You can see when you combine ICD pulmonary hypertension codes with PAH-specific therapy in the electronic health record, as well as a documented heart catheterization, you achieve the best positive predictive value with acceptable sensitivities and specificities. So I'll just summarize my presentation by saying I've tried to show that evidence from phase 3 trials is not completely sufficient to guide many clinical decisions. Pragmatic trials and real-world data can provide evidence on the effectiveness and safety of therapies under conditions of usual care. There are many challenges and barriers to the design and execution of these pragmatic trials, but they can and have been done. And electronic health records present opportunities to learn from usual care, but they too will require a lot of work on our part. Thank you.
Thank you very much, Greg. Very, very exciting comments. Well done. So the next speaker will be Isabelle Durand-Zaleski from Assistance Publique Hôpitaux de Paris in France. And she's going to discuss is PAH therapy getting too expensive? Health economics and cost effectiveness valuation of PAH trials. Thank you, Isabelle.
So hello, everybody. I'll be talking about health economics, and this is a topic that already has been mentioned several times in this conference regarding either pulmonary hypertension or cardiovascular disease. The first thing I would want to do is discuss the misconceptions about health economics. So what you should not be expecting from health economics study. Several misconceptions regard the fact that health economics is viewed as a tool to reduce healthcare expenditures. Just be aware that in none of our countries, so far, health expenditures have been reduced ever since we were born. So the issue is not reducing healthcare expenditures, it's making sure that we get what we want from the money we're spending. And health economics normally should be doing that. There's also been talk about the high cost of drugs, and actually this was somewhat the title of this presentation. Is it getting too expensive? Again, it's a very heated debate in the world. Some argue that high costs, and we're also talking about gene therapy and CAR-Ts in oncology, reflect the high costs of R&D. To me, it's slightly amusing that people would use this argument to justify high costs, because normally when you say that prices reflect production costs, it means your frame of mind is that of central planning, which existed in former communist countries. And so I find that quite disconnected from the reality of drug manufacturing today. The last misconception is, and that I often find that even amongst my colleagues, is that an economic evaluation means a cost calculation. So it's absolutely not true. And this possibly would be the one sentence I would like you to remember from that talk. An economic evaluation is not a cost calculation. It's trying to find how much health do you get from the money you are spending on healthcare or on a new therapeutic strategy. Sorry. So what we're doing is building economic evaluations alongside clinical trials most of the time. I'll give you an example of something different later. And the general idea is that we want to know how much money is spent on each arm of the trial. And that's usually fairly straightforward. We separate resources into medical, non-medical, direct and indirect. And again, that's fairly straightforward, and it's a typology that every country uses around the world. And now what we do is exactly the way we calculate the difference in outcomes between trial arms, we calculate the difference in costs between trial arms. And then we relate one to another. So in order to do that, there are plenty of guidelines. And again, you're certainly not at loss when you have to do an economic evaluation alongside a clinical trial. There are lots of recommendations. And the most important here in this slide is that we absolutely need to measure quality of life using a standardized instrument. It's a questionnaire which is validated in about every language that exists in the world. It's a 5-question questionnaire, self-administered. And so it's not a burden on patients. Why are we using this? It's simply because, as we are discussing spending healthcare money amongst medical specialties, we need a common metric for the measure of health. And it cannot be survival or progression-free survival or adverse events averted, just because that would not be relevant in certain disciplines, like ophthalmology or rheumatology, for example. And therefore, there has been the broad use of this questionnaire, which is now accepted, despite its flaws, which have been extensively published, which is used as the universal measure of health. So this is what you should keep in mind when you're planning a health economic study, to use the EQ5D questionnaire. So, and also another important point, when there is an economic evaluation alongside a clinical trial, is that we need to have the same duration of follow-up for the entire patient population, because resources are calculated over a given time period, which often in trials would be called time horizon. And this time period needs to be the same for every patient if we want to do a proper cost comparison. And so one problem we've sometimes encountered is censoring, and this would not do for us. So I know it's an additional burden, but we need to have the pre-specified follow-up period for every patient in order to do a cost calculation that is valid. And so the result of an economic evaluation is just saying, how much do I have to pay to get an extra health benefit? It's not how much do I have to pay more. It's I get a health benefit. How much is that going to cost me? And this is what we call the incremental cost effectiveness ratio. And so it's a difference in costs divided by a difference in medical result or in health. And what we do is also analyze uncertainty. And I will refer you back to the talk of Eric Vico about Bayesian analysis, because this is pretty much what we do. We do not have a pre-specified hypothesis that the incremental cost effectiveness ratio should be below a certain threshold. What we do is look at the probability that it is over a certain threshold or below a certain threshold, but it's always in terms of probabilities. And so it's always a result that is given to the decision maker as, if you use this new drug, you have an 80% chance that the cost effectiveness is below a certain threshold. And so it's completely a probability. So it's rather close to the Bayesian approach that was explained earlier. So here are some examples of the results. So we calculate on the left, you see that we calculate the costs of a new strategy compared to an old strategy. And you see what elements would be included in the costs. And then on the left, what's called table 6, on the right, sorry, what's called table 6, you have the results. So what we have is that you have one drug, the cost associated with not the drug itself, but the entire strategy over a lifetime. And then what we call the QALY, which is health, and it's quality adjusted life year. So it's survival multiplied by quality of life. And what you have then the last column called incremental is how much more do you have to pay and what is the gain in health. And the graph that is called a tornado diagram, because normally it looks like a tornado, but in that case, it's a strange one, is the uncertainty analysis. So we vary each parameter and we see how it affects the final result. This is one way of doing an uncertainty analysis in economic evaluations. Now, another option, if you don't have a trial at hand, is to do a model. And this also is something very common in economic evaluations. So you take probabilities from the literature and you imagine two cohorts of patients undergoing different treatments, and you see how their health changes over time and how the costs associated with the health states change over time. And this is very useful when you have a short-term trial, which you need to extrapolate over the lifetime period of a patient. So most commonly now, both the trial-based data and the extrapolation using a model are combined to provide a result. And so this is, I'm sorry, some of it is in French, but this is what happens when you have 3 drugs to be compared. And so you see that on the bottom left, this is what we call the cost effectiveness plane. So you compare strategies in terms of costs and quality of life, and you see which strategies are doing better and how all here 3 strategies compare to one another. If you don't want to pay much, well, you take the absence of treatment, which is the blue one. Then if you are ready to pay a lot of money, because here you see that it's 200,000, but then you get 6.5 quality of life years, then this is what you should do. But here in that publication from the French National Health Authority, the red one is considered dominated, meaning that if you want to spend money, don't spend it on that strategy, better choose the green strategy. So in conclusion, it's not difficult to do an economic evaluation alongside a clinical trial. Just do it anticipated, so from the start of the trial. The requirements are here. Having survival, it's not complicated because you usually have it. Quality of life, do not censor. And then for the cost data, it can be drawn, and this is what you've heard just earlier, from claims, from diaries, and from interviews. And missing data is always a problem, but you are very well aware of that. So thank you very much.
Thank you, Isabel. That was a great talk on the health economics, and I thank you for clarifying the economics versus cost. With our last lecture, I welcome Valerie McLaughlin from University of Michigan, and she's going to tell us which drugs are ready for phase 3 clinical trials. And then at the conclusion of her talk, we will transition to an open panel.
Great. Well, hello, everyone. Good afternoon, good evening. And Mark and Marty, thank you for having me. So many of the comments in the talk so far today have been so germane for us thinking about the upcoming and ongoing phase 3 clinical trials. And in order for us to have plenty of time for a robust discussion, I'm going to keep my comments brief. These are my disclosures as they pertain to the area of pulmonary hypertension. And I'm really going to talk about trials not in much detail, but I'll mention some trials, old pathways, new formulations. There are a number of these trials ongoing right now. And the main goal for many of these trials is to improve patient convenience. These are therapies that we know are effective. They've already been studied in randomized controlled trials, but have side effects or very frequent administration or are more cumbersome. And so this is a patient convenience issue for some of these drugs. For example, the multiple formulations of inhaled treprostinil with various DPIs and other modalities, really not trying to advance efficacy, but really mostly trying to improve patient convenience. There's an ongoing phase 3 trial of ralinepag, an orally available prostacyclin pathway agent, which interestingly has an event-driven endpoint, and this is once a day and may provide efficacy and more convenience for patients. And of course, the inhaled SGC stimulators that Paul mentioned. I'll move on to what are some of the other novel agents. And Mark mentioned a whole laundry list of novel agents, and certainly I can't cover them all. Some of them are much earlier in development, but the one that I think is very interesting that is ongoing phase 3 trials right now is sotatercept. And as you all know, this is a novel mechanism of action meant to rebalance the pro-proliferative and anti-proliferative state in PAH, primarily through the BMPR2 pathway. And I think we were all very impressed with the phase 2 clinical trial that has already been published. And the phase 3 clinical trials, I think, are really interesting for this agent because it's not just one trial. It is a whole program of trials meant to target different stages across the spectrum of pulmonary arterial hypertension. And as you all know, phase 3 trials are challenged right now by many of the topics that have been raised: the patient selection, the background therapy, the efficacy on top of such robust background therapy in many areas, and how do we deal with that? And I think another question that we'll address in the discussion is, what is the bar for novel agents? Big endpoint-driven trials are obviously take longer time and are more expensive, and that could be a challenge for some of the smaller companies making some novel advancements. So this is the phase 3 development program for sotatercept. The STELLAR trial is the trial that is ongoing currently, which is looking at a primary endpoint of 6-minute hall walk in patients already, prevalent patients, primarily randomized to sotatercept or placebo. And of course, we expect that many patients who will come into this trial will be on very aggressive therapy, often double or triple therapy. And it's a 24-week trial. The Hyperion study is going to look at a different question, and it's going to address whether or not adding a therapy in someone who's been recently diagnosed but does not attain low risk status with the initial treatment choice would benefit from the early addition of sotatercept. So that is the Hyperion trial, which will look at intermediate or high risk recently diagnosed patients, and that will be a time to clinical worsening trial. And then the Zenith trial, I don't think there's anything that's been done like this before. It's really a trial to look at patients who are very advanced, functional class 3 and 4, high-risk patients, and it's a time to event trial that includes death, lung transplantation, PAH-related hospitalization. So it's an entire phase 3 development program, which I think is really interesting. So I want to mention a couple of other agents that are kind of phase 2B, kind of progressing to phase 3. I think imatinib has already been mentioned. We have some experience with this, and I think many of us were impressed by the efficacy, but obviously the approval of this was hindered by some of the side effects. And so this agent, AB101, is a dry powder inhaled treatment of imatinib. And the goal there is to have the efficacy of imatinib more directly targeted to the pulmonary vasculature with less systemic absorption, so that perhaps we have less side effects with just as much efficacy. And the phase 2B trial is ongoing, and the phase 3 primary endpoint will again be 6-minute hall walk after 24 weeks. And then I also want to mention seralutinib, which is another novel agent. It was really designed to affect some of the known pathologic abnormalities in PAH, including in the PDGF, c-KIT, and BMPR2 pathways. And again, the goal is to rebalance pro-proliferation and anti-proliferation. And there is a phase 2 trial ongoing right now of this agent, the TORREY trial, which is a typical phase 2 trial with pulmonary vascular resistance as the primary endpoint. Again, likely to be a prevalent, highly pretreated population. But I want to mention this because I think it's really important that as we do clinical trials, we continue to learn, and it's important to advance the science. It's important to start studying new endpoints in our current studies in phase 2 and potentially even phase 3 trials. And so this study is going to have a sub-study that looks at heart rate monitoring, as well as a functional respiratory imaging sub-study using CT. And then the last therapy I want to mention is rodatrostat, which again is another novel agent. We're so lucky to have so many novel mechanisms of action being studied right now. This will target the serotonin pathway, has already undergone quite extensive phase 1 testing, and currently a phase 2 study is ongoing in the US to look at rodatrostat in patients with PAH. So I want to leave much time for discussion. You know, all of these therapies that I mentioned, and of course, this is just a sampling, it's impossible to include everything, are going to be plagued by many of the topics that we've already talked about. So we're currently in a very trial-rich period, looking at advances in current therapies that are primarily for patient convenience, but also a number of novel mechanisms of action, either undergoing or nearing phase 3 trials shortly. So thank you.
Up next, the CVCT Forum multi-stakeholder moderated debate, Redesigning Pulmonary Arterial Hypertension Trials, chaired by Marty Gomberg-Maitland, Washington, USA, and Marc Humbert, Paris, France.
Hello, we are now together, and it's great to see all the faces. We have invited a lot of people, and it will be a challenge to have everybody jumping in, but obviously we will have a second multi-stakeholder debate after this one. So without any further introduction, I would like to thank all the speakers who have already shared their views. It was, I think, very exciting and very good. I'm going to ask a first question to Steve Kaut, and then Marty will take the lead, and then anyone who wants to jump in, please jump in. And if you are dissatisfied, you will have a second show in 2 hours. So Steve, good to see you in good shape.
Nice to see you.
As always, very sharp. So I know you have worked a lot on 6-minute walk distance, and of course this part 1 is more traditional, and the second part will be more eye-opener and innovative. But 6-minute walk distance, It has been a valuable endpoint in PH, and it has led to the approval of many drugs. It is a very simple endpoint. We can perform it almost everywhere in the world, and it is currently challenged by many, but it remains an approvable endpoint for many agencies, and it really reflects improvement. So Steve, would you consider that 6-minute walk distance will become soon a historical tool, or will it remain a component of combined endpoint for the next year? So it's a long question.
Yeah, no, it's a great question. Thanks again for inviting me. So it has served us well, right? Most of our therapies have been approved based on this endpoint, and those therapies haven't really disappointed us in clinical practice. They work. So clearly there's something about the 6-minute walk test, which is good, that it's reliable and somewhat valid. Does it encapsulate all parts of a therapeutic effect? I don't think so. Is it a surrogate endpoint? Most likely not. So again, we've published this, I think almost 10 years now, looking at whether the effect of a drug versus placebo on the 6-minute walk explain the effect of the drug versus placebo on outcome, and it really didn't. So as a surrogate, I don't think it's necessarily valuable. As an intermediate endpoint or a clinically important endpoint, it might be. Now, you mentioned the problems, Mark. It has to be done in clinic by a tech. So we're interested in two different things. We've actually done, I and Corey Mentitulo have done some studies of doing a remote 6-minute walk test. So giving patients some supplies, finding a course, measuring it out, doing it with a partner, and having the patient do the 6-minute walk locally or remotely where they are. And we have a paper in revision right now where it seems like that remote testing actually is pretty good at capturing what the 6-minute walk would be in clinic. And I don't have to tell anybody on this call why remote testing might be useful in this day and age when my patients don't want to come to clinic. The second other innovation, I think, is actigraphy. And I think this has been spoken about before. The 6-minute walk is great, but it reflects my patient walking up and down a hallway in my clinic. And that's not what my patients do at home. And that's not what brings them pleasure or makes them happy. They want to go to get the paper or play with their grandkids or go shopping or go on a trip. And so using things like actigraphy, which may better gauge how active a patient is, how much they're able to do in their home environment, might be the thing in the future to displace 6-minute walk. Sorry, a long answer for a short question.
That's what I was thinking. Anyways, I see that Veronica has her hand up, so I'm gonna utilize that and recommend that people do that. I am scanning all the pages because we do have a big panel to make sure that I see everybody's hands up. Veronica, what you had something you wanted to add?
Yeah, thanks very much, Marty. And I really, this was a very interesting discussion and presentation. So I appreciate being part of it. I think that as you discussed, that possibility of doing a withdrawal trial, it's fascinating to me. I think that with that, as I see it in the future, I see that a lot of patients come to us very sick, and we obviously try to throw at them everything and all the medications, parentals and everything. And then obviously we try to get them better, get their RV function better and everything. And I think we're at some point now that we need to start thinking about withdrawing, because I think that will be a lot more appealing to patients. They will be able to, okay, I do all these, and then when it gets better, I know I'll get off my pump, maybe to a tablet. And if we can find strategies, I think that the biggest limitation in my mind is who's going to fund that study, because obviously it will be withdrawing one of their medications. But so as I wanted to bring it up to the panel as this, if we design a trial like that, how will we get it funded? Because obviously most of the companies are invested and they don't want to withdraw their medications, right?
Yeah, I think it's a great point. And I think that when I initially started prostacyclin use 20 years ago, we didn't have all these options. And now many of those patients are off of their IV on multiple combinations. And when is the right time, and how do we do that best? I think that Greg's talk may have touched upon the pragmatic real world data could be how we do that, and having centers that are focused in that. I agree with you that cost is going to be difficult. Dr. Preston, I see you have your hand up, but I think we should just ask some of the industry folks, is this something that you guys are interested in? Does it need to be in a randomized withdrawal kind of a trial base? Has anybody ever thought that this could be something that they could pursue? My guess is that that's not going to be their focus, but it's something that we need to do as a community. Isabel.
Yeah, well, we have government-funded studies which often look at tapering. And so there would be non-inferiority trials. I'm not sure whether Eric has already been involved in one or several, but so there would be non-inferiority trials, and there would obviously be a cost analysis as well, but these would be government funded.
Yeah, Marty, Marty, I thought immediately this is one for post-approval, pragmatic cluster design trial, perhaps. You could identify with whatever parameters you chose the population of interest. You could presumably even randomly assign them. And the main thing is to make it large so you get high quality data and efficient. Keep the design as simple as possible with a pre-specified endpoint. It's a great example of a pragmatic trial that likely wouldn't be funded because it generates perhaps some savings for insurers and people. It also generates some quality of life for patients, which is a reason to do it. So I think it has value, and it strikes me as a great opportunity for a post-approval randomization trial.
I had called on Ben, but I don't see him on the screen.
Joanna wants to speak.
I've raised my hand again.
Yeah.
Oh.
Hey, Marty. So an honor to be here on the panel with all of you, kind of putting some of these ideas into practice. Val highlighted our trial at Aerodate Therapeutics. We're doing an adaptive 2B3 trial now with a long-term open label extension. We talk about randomized withdrawal trials all the time, and we're not afraid of spending some money on it. We've been in discussions with regulatory agencies about what it could look like, and with folks like you. And I'm really glad to hear people that, coming up in this panel, that people want to pursue that more. Because before, I think a lot of times there was fear of taking a drug that's working away from someone, not just from a sponsor like us, but also from doctors, right? If the drug is working, taking it away can be a drastic thing. And we've had some bad experiences in the past. But I would not say the default view from our perspective of industry is that there isn't money to do one. I think it's about doing it smart. And as you highlighted, Marty, in your presentation, convincing ourselves that it's safe first, right? And that we have a good biomarker or something that will guide how we actually put it into practice.
Okay, so Awana.
Hi guys, such an amazing session. I'm really enjoying it. So about withdrawal trials, let me make a comment that back in the day, we were all very wary of participating in this type of trials, although FDA had some mandates. And when the withdrawal treprostinil trial happened, my center decided not to participate in it. And we know all the results. However, now we do have so much background therapy that I think, as the prior speaker said, doing it smartly and having a very low threshold to switch back, I think it can be done. If you look at the temporary interruptions of selexipag in the Griffin trial, a post hoc analysis, it seems that it was safe, even though it was not warranted or planned in those folks who interrupted their drug. And I think it's mainly because their background therapy, prevented them from slipping. However, as PH experts, we do have experience of transitioning from more invasive infusion parenteral therapies to less aggressive via oral or inhaled therapies, and we have not had full success. So I would caution, if we design this kind of trial, to be absolutely careful and maybe aim for a low bar, such as a BNP level, not full with other markers, but be very vigilant. But my question is, let's switch gears to quality of life and economics talk, which was absolutely amazing for me, at least, because I've learned a lot. EQ5D questionnaire, which is a global health questionnaire, does it need to be validated in your particular disease that you are studying before you implement it in a clinical trial? If you could give us a little bit more details about
that's that's its strengths and major weakness regarding what clinicians would expect. So it's a generic questionnaire, pretty much like the SF-36. It has 5 domains, hence the 5D, 5 dimensions. So it's mobility, self-care, social life, anxiety, depression, and pain. And so, and it's on a scale of either 1 to 3 or 1 to 5 in the newer version. And you understand very well that its purpose is to measure health regardless of the condition, because its purpose is to allocate money, which has no dimension, amongst medical specialties. And so you have to have a tool that does the same. And that's why it was created. Its weakness is that it's not very sensitive, and therefore we're discussing the 6-minute walk test. You could gain 50 meters or 60 meters. It's not obvious that it will translate in an important change on the EQ5D, unless it really changes your social life, because it allows you to do something different. But then the impact on the EQ5D would be likely very small, and that's its weakness. And that's why in clinical trials, the general opinion is always to combine a disease-specific instrument like the St. George or any other to the generic instrument.
Mm-hmm. And has it been sensitive enough in clinical trials in orphan diseases? It may have been in large cohorts of heart failure or cancer or other, conditions, diabetes, but is there any data on orphan diseases where clinical trials typically enroll less patients than the more common disorders?
So patient numbers is obviously an important element because it increases uncertainty on the results. So that's a common factor. It's also true for costs, so and also true for outcomes. So I don't think it's any different. Now, yes, the EQ5D has been used in orphan disease trials. Again, there has to be a major change so that it would show up. More interestingly and innovatively, there's discussion on whether and to use it also on carers, and whether it is appropriate to aggregate the scores of the carers and of the patients. And so this is another current topic.
Thanks. I think it's tricky because it's with such a small difference, you're not going to see it. And so I'm not sure that that's going to be the perfect one for us in the trials that we're doing. I just have a question from one of the folks in the audience, which was, a lot of the older therapies now are becoming generic, and we don't have any generic clinical trials. How are we supposed to evaluate generic formulations? Which I open that up to anyone. It's not an easy question, obviously. And I think that we've all found that sometimes the generic is not as good and sometimes it is. I don't know, Valerie, do you have any insight into that?
Well, I mean, just the experience that we've had, we sort of vehemently opposed when the FDA approved generic epoprostenol. And, when I was very active in the PHA at that time, and we went to them and they basically said, it's bioequivalent, it's, directly IV. And they, I think, have their methods for evaluating the bioequivalence of the drug and have allowed it to be approved that way. I think we all have experience with generic drugs at this point. I guess my experience is, for the most part, most of the patients do okay. Every once in a while, you have someone who insists that they're, very different on a generic, and you try to get them the brand back. But I think many of our patients tolerate generics just fine.
Yeah, I think sometimes there might be slight differences in dosing that you can sort of tweak. Antoine, I see you have your hand up.
Oh, yes, thank you. Thank you. A fascinating talk. And I have a question which is probably on the other end, not on the generic end, but on the front end. And I come from a background of cancer, and I've been blessed by putting Tagrisso and HER2 olaparib, on the market. Obviously, we have a path to approval, which is probably faster and different from what you're experiencing here. So my question is for Valerie, which is, how about, you've described phase 3 plan. It's very striking to me that sotatercept, is very reasonably likely to demonstrate clinical benefit. And it's on the back of a biological plausibility, which is extremely strong. So in my mind, this is something which should be approvable on the basis of existing data. And then the phase 3 design and the phase 3 program should probably be actually more post or peri-market and post-market, as opposed to a series of phase 3 randomized clinical trials, which are heavy and carry all the costs and the uncertainty and the difficulties. So what do you guys think about being more challenging in actually going after a quick, fast to market approval in a very well-defined and enriched population, and then have a larger program which is less RCT heavy and more, peri-market, as was presented, by Greg, the post-approval trial, essentially. So how likely are you to essentially, and obviously I come from Centessa, and I'm very interested with the BMP9, which is essentially a very disease-targeted, disease-modifying agent, just like sotatercept is. And thus, I would apply my experience to this future. So quick question for you guys. What is the appetite for actually changing the conventional thinking towards more aggressive?
Yeah, I mean, I'm happy to make a couple of introductory comments. There are many individuals who are on the steering committee for sotatercept on the call right now, as well as the sponsor. And, I guess in my heart, I feel like this is really an attempt to make a contribution to the field, right? They could do a 12-week or a 24-week 6-minute hall walk trial, get the drug on the market, and then let the rest of the stuff happen, or not, phase 4. But I think the whole group felt very committed to determining what role this therapy could have across the phases of the disease. Thought there were a lot of unanswered questions, particularly in those early diagnosed. Maybe everyone doesn't need triple therapy, but we know that a large proportion of the patients who start on ERA-PD5 don't get to where we want. Like, we know that if they're going to get there, we're going to see it within 6 months, and if not, we need to do something more. So that's a whole unanswered question. And of course, like the very end stage of the disease, it's been really challenging to enroll those patients in clinical trials. So I feel like it's an entire program that was designed with the sponsor and the steering committee to really make a contribution to our disease state. Janeth is here, Mark, Marty, Marius, Iwana, there's a lot of people.
No,
yeah, I was going to pass
don't
it.
get me wrong.
Antoine, I was going to pass
this program. I applaud this program. This program is terrific.
Antoine,
I
think
saying that the front end of it is can it be faster?
No, understood. And I think that, we want to see disease modification. The original study, right, the phase 2 is a longer study. It's not a 16-week trial. I think that we as a community need to do better. And a lot of times when we do a fast approval, there's so many patients that aren't able to get the drug because they're not in that class. And so this is, I think that we as a community said, these are the focus and we really want to get as much out of it. But Janeth, I'm going to call on you because you are at the center of it.
Yeah, thank you, Marty. And all of you in the steering committee too. I think we've been working very closely also with the regulators, Antoine. The difficulty, I think, it's what Marty already said about access. If you have a trial that it's very fast, very small, very quick, you might get a very small population that would have access to it. And also there is the challenge of regulators and also the reimbursement agencies and payers that they feel, even though the data may be very compelling on a small number of patients and the mechanism of action makes all the sense, that there is always that desire for confirmatory trials, for confirmatory data to really bring this to fruition. And like Marty and Val already said, we designed this in a way that we may have, these trials will read sequentially. So the first trial should read out, in probably in another year or so. And then after that, the other trials will continue to recruit and continue to deliver on data. So it's a sequential delivery of data that hopefully will expand the access and the label to as many patients that could be eligible as possible. But it is, even though we had the breakthrough designation in the US, we had the prime designation in Europe, it is difficult, I think, for the regulators because of their responsibility, budget-wise, public health-wise, to make a decision in approving a small trial or in an endpoint that may not be what they would like to see. For example, in our phase 2 trial, it was a PVR endpoint, and even though they acknowledged the value of the endpoint and the improvement that was demonstrated, they still are there's the thing about the approvable endpoint, the validated endpoint. And I think they feel it very difficult to deviate a lot from what is their policy and their procedures that are, standard.
Bjorn, I know.
Thank you for blazing the trail.
Yeah.
Thank you for letting me in. Very interesting session. I'm new to this community. Basically, I'm CMO of Cereno Scientific. I've been in research for 40 years, but in other areas in cardiovascular. And my question is really, when are the PH therapies going to be compared to each other? I mean, I see most studies being versus placebo, and then we get into the arsenal. But when I'm used to that, you have to prove yourself to existing therapy or to be how you place yourself or how you place yourself as a starting therapy versus the others. That so I'm interested in some opinion on that.
Do you want to take that, Mark? I mean, I can answer it.
Well, I just offer again, that's a great opportunity for post-market pragmatic trials. And you could just envision, as an example, comparing one endothelin receptor antagonist to another with a large cluster randomized design where you randomize by center.
Yeah, I think.
And I would only say if there's appetite for such studies, there may or may not be.
Yeah, Greg, I think that the more pointed is that it is an orphan disease and we have a lot of therapies and we debate on which therapy we should actually go at. Like, how do we know which phase 2 to enroll our patients in? And we don't really have that understanding yet. I think we're a little behind other disease states as far as true biomarkers and ways to target, and that could be part of it. But to do a big non-inferiority study, to me, it's just patient populations to compare existing. Yeah.
Yeah, Marty, it's a great question from Björn, and I think right now we have standard of care, and right now for incident patients, very many patients are initial dual combination therapy. But you are right that after this initial phase, there is a variety of options, and we can compare them face to face. It's more academic research than usually industry-sponsored research, but with Olivier Sidbon, who is connected right now, we have thought about that, and we try to push for that. And of course, novel agents like sotatercept, but also others, could be compared with another approved option. So that's, I think, a good point, and we need to go to that direction. So thank you, Marty.
Sure, we have less than a minute. I will say that in the heart failure trials symposium yesterday, they talked about doing diuretic versus another agent. So I think that everybody's sort of thinking the same thing to our actual standard of care. Should that be standard of care? I see Paula and Brad, if you guys have 10 seconds, I think they're going to cut us off.
I have a very quick question. We're dealing with the aging population, patients with multiple comorbidities, and the trials are done on background therapy. What are the, in terms of dose adjustment, which we do in real life, because these drugs may have additive or synergistic effects, but not necessarily at full dose. And do we have any plans in addressing this question?
I think we're going to have to continue that on the next one. Brad, do you have a nice?
You will not be cut off. Keep going if you have. But I can see you are real diehards, because there are one further session to go on the same topic. I am so pleased that about the engagement and excitement. So keep going if it is your wish.
Yeah, sure. Anybody want to take that question?
I think the multimorbid population is a very important one, and they truly deserve dedicated studies. What I don't like is when companies want to have very clean population, which are very different from the population seen in real life. So I take your point, Paul. We need to consider patients with multiple comorbidities, and Nazareno in the guidelines and in the World Symposium has always considered that patients with comorbidities correspond to a population which require more well-done studies. So your point is well taken. And I think Brad raised his hand, and Isabel, and then we will pause, I think, for a couple of minutes. So Brad, maybe you are muted.
Thank you very much.
Yeah, good.
Thank you very much for the invitation. A very fascinating panel. I think it was Marty who talked a little bit about the historical roots of clinical trial engagement in this disease, which, as she pointed out, focused really on salvaging mortality in patients with end-stage disease. And so my question to the panel is that, of course, we've progressed beyond that now, and whether the clinical trial endpoints should really match the stage of disease, focusing more on early detection of patients and how those endpoints may be catered to different needs compared to those who are enrolled with more severe symptoms, that survival may be the principal target. Thank you.
It's a great point again. It's a very good point. I don't know, I can't hear anyone speaking, so I would jump in and say to Brad that we have room for that indeed, and we speak a lot about that. Early detection together with early intervention would be, I think, a very smart approach. We have all the technology right now, and we have some populations at high risk of developing PH or PAH. It's always the big question, but early intervention, we have room for that, but also enrichment for more events, traditional events, was, I think, the focus of Marty's presentation. So we need also to think about early intervention and which endpoint should be suitable for that, and how can we make a meaningful randomized control trial within a limited time. That's always a challenge in early disease.
Yeah, and I think that we could focus on the diagnostics. I mean, all of our patients are getting CT scans to look at the lung parenchyma and to look for clots. And I think that as the technology is improving in that, there's a paper that's going to come out looking at the RV and how that picked up RV dysfunction 25% of the time that it wasn't seen on echo. So I think that, there are going to be better ways to find those patients at risk that we could focus on, because I don't think it's a long-term study looking for the development of the disease that we need to do, but maybe just better take the data that we have and pick up patients sooner. And then you said Isabel, yes?
Yeah, just an answer, a comment on the comorbid patients. I think they should be studied, but I think we should be aware that the results are likely to be disappointing from experience, just because of the competitive risks. And so we did some pragmatic studies, taking every person in a center, and then there's so much reasons that these patients would need healthcare, that it's very unlikely that one single intervention will prove beneficial and will help reduce costs. For example, this was telemonitoring, for instance. And that's something we have to understand and to accept, that when you do a study on high-risk patients, it's completely different from the usual RCTs, and you're very unlikely to show that a single element of care will change something. And it's pretty hard to swallow usually by the investigators.
Isabel, amazing comments. And that brings me to the thought of maybe providing a holistic approach to these very complex comorbid patients. And the way we have the REVEAL-2 score for, risk calculator, maybe we should have a holistic several parameters approach to improve patient well-being in the group of patients with pulmonary hypertension.
Thank you. I think we should all take a break. For us, it's lunch here on the East Coast. I'm guessing you guys are at a pre-dinner snack in Europe. And then
cocktails.
We can.
Yeah.
So we and Dr. McLaughlin and Dr. Cereno will be moderating, co-chairing the next session. And thank you all for joining us and being a part of this vibrant panel.
Thank you.
Thank you, Marty.
Up next, the evolving landscape of pulmonary arterial hypertension phase 3 trials. Part 2, redefining clinically meaningful endpoints, chaired by Valerie McLaughlin, Ann Arbor, USA, and Gérald Simonot, Paris, France.
Well, hello, everyone, and welcome back for our second session on pulmonary hypertension, the evolving landscape of pulmonary arterial hypertension phase 3 trials, part 2, redefining clinically meaningful endpoints. On behalf of my co-chairman, Dr. Gérald Simonot from Paris, I'm Valerie McLaughlin, and I would like to welcome you to the session. I'm really looking forward to it. We had such a great discussion at the end of the last session, which I know will continue. The presentations will be very brief, so we have continued time for discussion. And to start us off, it is my co-chairman, Dr. Gérald Simonot from Paris, talking about a critical appraisal of classical endpoints in past and recent PAH phase 3 trials, 6-minute hall walk and time to clinical worsening. Dr. Simonot.
Dear colleague, it's my pleasure to introduce this second session. My talk is on the critical appraisal of classical endpoint used in the past and recent phase 3 trial for PH. Here my conflict of interest disclosure. Today, more than 10 drugs have been approved for the treatment of pulmonary arterial hypertension. The main characteristic of the primary endpoint in a registration trial is to be a clinically meaningful outcome measure of how patient feels, functions, or survive. Between 1996 and 2013, in 8 pivotal trials, a measure of exercise capacity, the 6-minute walk distance, was chosen as primary endpoint. The first trial were we can find a 6-minute walk distance as a primary endpoint was published almost 30 years ago by our nice colleague, Robin Bast. Unfortunately, passed away 8 years ago. In this trial, where compare the effect of prostacyclin versus conventional therapy. How the steering committee decided to use the 6-minute distance as primary endpoint. In fact, the steering committee initially suggested hemodynamic as a primary endpoint, but FDA insisted that an acceptable primary endpoint needed to either to be a measure of patient symptom, exercise capacity, or survival. Sponsor was unwilling to choose survival and was reluctant to use functional class because considering too subjective. As a result, a measure of exercise capacity was chosen and 6-minute walk test was preferred to the treadmill test due to overrepresentation of a pulmonologist versus cardiologist in the steering committee. Between 1996 and 2013, 8 phase 3 pivotal trials have used 6-minute distance, leading to the approval of many drugs. As you can see here, this trial has a short duration of between 12 weeks and 17 weeks. The sample size is between 80 patients to 400 patients. And what is important to observe is the first trials were performed in patients naive of background therapy at inclusion. And at this time, the improvement in the 6-minute distance was between 50 and 60 meters. For the 2 last trials with tadalafil and riociguat, some naive patients represent only 50% of the population, and many patients are on background therapy, ERA, or oral prostanoid. And now the improvement was lower, between 20 to 30 meters. What are the advantages and drawbacks of using a single distance as primary endpoint in PH randomized controlled trial? Pros is simple, inexpensive, reproducible, valid tools for assessing exercise capacity, accepted by regulatory authorities for registration of PH drug, both in US and Europe, and initially considered as a reliable surrogate of outcome. But there is some drawback, less improvement seen in patients without advanced disease with a ceiling effect, especially above 400 meters at baseline. Improvement is attenuated by inclusion of patients on background therapy with less effect. And today, in addition, there is growing evidence that a single distance improvement is not a reliable surrogate of outcome. Now we have some data from observational study and randomized controlled trial in PH suggesting short-term change in 6-minute distance at 6 months are not associated with a better prognostic at long term. Whereas absolute change, absolute value at baseline, and especially more than 400 meters, is associated with a better outcome. After this first trial, we moved to morbidity mortality trial in PH between 2013 to 2020. In fact, the term of morbidity mortality is not clear, and we do have to use time to the first event of clinical failure as primary endpoint. And as you can see, 4 trials with a large population between 600 to almost 1,200 patients, with a large period of inclusion around 3 years, but from 1.5 to 5 years, and a maximum follow-up of 5 years. For the first trial, the trial of macitentan in PH. It is a double-blind placebo-controlled study comparing 2 doses of macitentan, 3 and 10 milligram in 700 patients. And at inclusion, 61% of these patients were in PD-5 inhibitor. As you can see here, 10 milligram macitentan is better than 3 milligram and is better than placebo. And this positive primary endpoint with less event with 10 milligram is driven by the worsening of pH with 24% with 10 milligram versus 37% in the placebo group. There is also very few events for prostanoid initiation and lung transplantation. And regarding death, as you can see, 6.6% with 10 milligram versus 6.8% in the placebo is similar. The second trial, the Griffon study with selexipag, is a double-blind study comparing selexipag with a maximum dose of 1,600 micrograms twice daily in almost 1,200 patients. And at baseline, 47% were on PD-5 or ERA, and 30% was of double combination therapy, PD-5 and ERA. And here again, the primary endpoint was positive. We have less clinical worsening event in the selexipag group as compared to placebo. But this effect is driven by hospitalization. 13% was the selexipag group versus 18% in placebo group. Disease progression, 6% in selexipag versus 17% in placebo. Regarding death, is similar, even a little bit higher in selexipag, 4.9% versus 3.1%. Again, for initiation of parenteral prostanoid and lung transplantation, very few events. A very interesting study has also used this endpoint. It was a study of ambition, use of upfront combination therapy with ambrisentan plus tadalafil versus either of this drug in naive patient, naive of treatment. Is incident patient. Is a double-blind study. And as you can see here, the combination therapy is better than the pool monotherapy group. But again, this effect on the primary endpoint is driven also by hospitalization, 12% for the monotherapy group versus 4% for the combination therapy group, and also for disease progression and for unsatisfactory long-term result. But here again, there is no difference in terms of death, 3% for monotherapy group versus 4% in the combination therapy group. The most recent trial was performed with oral treprostinil. It is the Freedom EV trial comparing maximum dose of oral treprostinil 12 milligram TID versus placebo. Here again, a lot of patients are on background therapy and inclusion. And the effect is positive on the primary endpoint of time to the. Sorry for the technical problem, but I continue with the Freedom EV study. And as I told you, this study is positive, less clinical worsening event in the group treated with oral treprostinil. But again, it's mainly due to less disease progression, generally considered by deterioration of functional class and by deterioration of the walking distance by 15%. And as you can see here, again, there is no difference in terms of death, 4.1% versus 4.3% in the oral treprostinil group. Why mortality is not improved in all the recent PH trial using time to the first event of clinical failure? Is relatively simple. In fact, in this disease, death is rarely a first event, and sudden death before any sign of clinical deterioration is extremely rare in PH as compared to left heart disease, for example. In addition, inclusion of patients very severe in functional class 4, very high risk patient is rare. Generally in trial, less than 2% of patients are in class 4. For the less severe population, functional class 2 or 3 of phase 3 patient, representing the majority of patients included, these patients are in fact stable because in the protocol, we need to have at least 3 months of stable treatment and no recent hospitalization for at least 3 months. And it's difficult in this case for this stable patient to have a death very rapidly observed. In addition, the sample size needs to be very large, and PH is a rare disease. However, we observe a positivity of the composite endpoint, especially the hospitalization due to worsening PH and or disease progression. But this endpoint is, in fact, very clinically meaningful endpoint. And my co-chair, Valerie McLaughlin, recently published an analysis of Seraphin and Griffin study, studying the clinical worsening event occurring in the first 3 months and showing that higher is the rate of worsening event during the first 3 months, higher is the risk of long-term mortality. And is observed both in Seraphin and in Triton study. In summary, between 1996 and 2021, 14 drugs have been approved for the treatment of pulmonary hypertension. It is an incredible achievement for a rare disease. Between 1996 and 2013, these agents were approved on the basis of improvement of the 6-minute walk distance at 3 months. However, in more recent trials, efficacy of new drugs have been assessed as add-on therapy to background therapy with lower increase in the 6-minute walk distance, generally less than 30 meters, than study in treatment naive, making interpretation more challenging. In addition, it is now well established that the improving in 6-minute distance at 3 months is not correlated with the long-term outcome. More recently, the primary endpoint moved from 6-minute distance to time to the first event of clinical failure, leading to the approval of several new agents. However, this primary endpoint has some drawback. It requires long-term trial and large sample size. Among the different components of clinical failure, mortality is never improved as compared to placebo. Lastly, it does not provide some information on clinically relevant endpoint of improvement. And during the next presentation, we will do an overview of the possibility of new endpoint for this phase 3 trial in PH. Thank you for your attention. Look, now it is my pleasure to introduce the second speaker. The second speaker for this session is Marius Hopper from Hannover University Hospital in Germany. And his presentation is a proposal of novel endpoint based on multiparametric assessment risk stratification.
Hello, Val, Gerard, everybody. So I continue here with proposals on new endpoints based on multiparametric risk stratification that have already been used in some clinical trials in our field. disclosures first. And then, I mean, I'm just recapitulating some of the thought that have been already expressed here. Where are we? I mean, this 6-minute walk distance was just very nicely presented by Gerard. It's still accepted, and we know why. But we also understand that these improvements do not predict better survival. Time to clinical worsening, I think Gerard just pointed out the most important limitation this year, that it does not capture patient-relevant improvements. And that is the main treatment objective in our field. And then, of course, the large sample sizes, long trial duration. So definitely not perfect. Multi-component improvement endpoints have been used in the replace study that was mentioned before, and in Pulsar, I will come back to that. They have been proposed to the FDA, but they have not been accepted by the FDA as a primary endpoint for an ongoing study. And I think we will discuss the reasons later. And then, of course, risk stratification tools, they are also on the orbit here as potential candidates. And I will come back to this, and this will also be covered later by Dr. Benzer. So I think the key considerations here are that we certainly need an improvement endpoint that is relevant to patients. And I think 6-minute walk distance is doing that, but also carries prognostic information. A 6-minute walk distance is not doing that. And if we use a multi-component improvement endpoints, the FDA would require that all components must be relevant to the patients. And here is where we have a problem. So I hope you can still hear me. I think otherwise I will hear from you. So the multi-component improvement endpoints have been used in the replace study. There was a study switching from PDE5 inhibitors to Rio that Steve Cabot discussed earlier today, and Pulsar, the pivotal, not yet the pivotal, but the phase 2 study with sotatercept. And in both studies, the components of the multi-component endpoints were change in 6-minute walk distance, NT-proBNP, and functional class. A functional class is basically no-brainer. It's an improvement here. You maintain a low functional class. 6-minute walk distance studies choose improvements of more than 30 meters and some other variables here. And NT-proBNP, the threshold was set at 30% change from baseline. In both studies after 24 weeks. And the difference here was that in replace, 2 out of 3 criteria had to be fulfilled, whereas in Pulsar, all of the 3 criteria had to be fulfilled. And both studies succeeded with this endpoint here. And it was the primary endpoint in replace, and it was significant. And also in Pulsar, it was a secondary endpoint. But you see there was a substantial difference here. I mean, it's a very strong signal and also quite relevant here. The problem is that we do not know if this translates into better outcomes, because replace didn't have a long-term extension, and in Pulsar after 24 weeks there was no improvement any like clinical worsening, or not speaking of mortality, and there's an ongoing open label extension, but then all patients have transitioned to active drug. So long-term outcome based on this endpoint, this is something that is fully unknown yet. So what I'm presenting now is some analysis that we've done very recently on our Compera registry, and this is data that has been submitted for publication but has not been presented before and has not yet been published. So what we've done here is we try to mimic the trial situation. So we selected treatment-naive patients with PAH who have been diagnosed over the past 10 years, and those patients were selected for whom functional class 6-minute walk distance NT-proBNP were available both at baseline and at the first follow-up. And then we determined changes from baseline to follow-up, and based survival analysis based on these changes, making the first follow-up as the landmark for further survival analysis. And then we looked at changes functional class 6-minute walk distance NT-proBNP. We used the replace and Pulsar multi-component improvement endpoints as they were, but also with some modifications, and we used some of the established risk stratification tools. So using these inclusion-exclusion criteria, we were able to identify about 600 patients newly diagnosed PAH, for which we had all the information at baseline first follow-up. the median interval between baseline and first follow-up was about 4 months. And after first follow-up, we had a median observation time of 3 years. So I think this is quite relevant because this is something you usually don't have in clinical trials. And during this period, about 1/3 of the patient died. There were a few patients who underwent lung transplantation, were censored at the time of transplant, and we had 5% of the patients who were lost to follow-up that were also censored at that time. So what did we find? First of all, functional class, changes in functional class, they predicted outcome. So if you improve functional class, you certainly had a better long-term outcome. And this criterion was met by about 1/3 of the patients here. And when we were looking at change in 6-minute walk distance, there could be absolute changes or relative changes. And the same was true for NT-proBNP. We basically saw nothing. So this confirms previous publications and observations that at least when it comes to 6-minute walk distance improvements, it has now predictive value when it comes to consecutive survival. But also when we look at NT-proBNP changes, and especially the changes here, the more than 30% improvement from baseline that have been used in the 2 studies I mentioned, it does not translate into better survival. So the clinical value of this 30% improvement is totally unknown. It's an arbitrary value, but it seemingly doesn't really work. However, when we then look at the multi-component endpoints, and this is replace, and remember that in replace it was required to have 2 out of the 3 criteria improved, then if this endpoint was improved, then the long-term survival was significantly better than if the endpoint was not improved. And importantly, this endpoint was met by more than 50% of the patients. And if we use the modified replace endpoint, using any functional class at baseline, because replace included only functional class 3 at baseline, we even had more than 60% of these patients meeting the criteria. And again, it had prognostic value. PULSAR, also quite a strong signal. So if you met the PULSAR improvement endpoints for all 3 components of improving functional class 6-minute walk distance, NT-proBNP, you also had a significantly better long-term survival. And here again, we did the pure PULSAR analysis that included patients in functional class 1, 2, or 3 at baseline, and that was statistically significant, but it was met only by 24% of the patients because all 3 criteria had to be met. When we used any functional class at baseline, significance was still met in terms of consecutive survival, but again, only about 1/4 of the patients met these criteria. So finally, and this is a bit extending now in the next talk given by Ray Benzer, we also looked at the implications of an improvement in the established risk scores. So here we use the ESC-ERS 3-strata and 4-strata models, the French non-invasive model, and a modified version of REVEAL Light 2. We had to modify it because in Compera we have almost no BNP, but mostly NT-proBNP values. So we had to play around with this a little bit. What we found is that there were quite a few patients who met the criteria of improving these endpoints. This was ranging from about 40 to about 50% with the different criteria. And whenever you improved a risk, you also improved your consecutive survival quite substantially. So these differences were highly significant. And I just want to point out the French model here, which was the improvement criteria met by about almost half of the patients. And there's quite a big difference in survival. And when we did some Cox proportional hazard analysis on these endpoints, looking at the hazard ratio, if the endpoint criteria we met, that then we found that more than 50% reduction in consecutive mortality were seen with the PULSAR and modified PULSAR endpoint. But again, only about 1/4 of the patients met these criteria. We saw this with the ESC-ERS 3-strata model, and this was met by about 40% of the patients, and with the French model, so quite a strong signal here, and this was 46% of these patients. So there seemed to be a candidate here that might be quite attractive for future endpoint consideration in clinical trials. So I'd like to conclude from these data and also from what has been said before that I think it is clear, it's very clear that improvements in 6-minute walk distance NT-proBNP, they have no or little predictive value for consecutive outcome. And especially for improvements NT-proBNP. And this is a problem when we discuss these multi-components endpoints that I've shown you as endpoints of clinical trials. The NT-proBNP, there's no patient relevance threshold, at least none that we know. And therefore, it's very unlikely to go on to be accepted by the authorities. The multi-component improvement endpoints, they were met by a variable number of patients, but they do carry prognostic information. But again, the NT-proBNP component, as it was used here, is not validated and probably not patient relevant. Improvement in risk, and this was met by a sizable number of patients and carries prognostic information, and this also has been validated in numerous studies. I think there's no doubt about this. So I think what we need to do, and this is maybe a good starting point tonight, we need to discuss with the stakeholders, and these are of course the authorities, physicians and patients, if and how we can establish multi-component improvements and specifically risk stratification tools, which I think are the most promising candidates here as surrogate endpoints in clinical trials. With that, I thank you very much for your attention.
Marius, thank you. That was a really fantastic talk, and thank you for sharing that new information with us. It's quite relevant to our discussion today. So thanks, I really enjoyed your talk. We will now move on to the next lecture, which is being given by my dear friend and colleague, Dr. Ray Benza from The Ohio State University in Columbus, USA. He is going to talk about validated risk score as a primary endpoint predicting mortality.
Good day, everyone. It's really a pleasure to be here, and I want to thank the moderators, Dr. McLaughlin and Dr. Cereno, for inviting me to speak this morning on validated risk scores as a primary endpoint for future clinical trials. I think as we go into this, we must review a few of the basics, primarily what the performance expectations of an endpoint should be. And as we all know, and as reviewed in the prior studies, we know that a good endpoint for a clinical trial should reflect how a person functions, feels, and survives. And I'm going to demonstrate to you how risk scores do each of these things. A good endpoint should also be validated, ideally prospectively, and also be highly discriminatory so that we know that the predictions of outcome are sufficiently robust to represent the hard endpoints that we really are interested in. As I mentioned, a good endpoint should meet all the characteristics that I mentioned also, but in my conversations with the FDA and other regulatory authorities, we must make sure that a risk score appropriately reflects the connection between a new drug and a hard outcome. And what I mean by this is that a new therapeutic should change a score, and then the change in score should correlate with the change in outcome. So let's look at our contemporary risk scores that we have right now currently, and we won't take too much time going over this because I think everyone knows this. We have a variety of contemporary risk scores, two deriving from Europe. The Compare and the Swedish made use of the risk table and as a summated score. The French algorithm, as you know, uses a number of low risk categories that can either be invasively or non-invasively derived in order to determine the number of low risk categories. And then an updated portion of Compare using a more finer risk stratification and finer refinement of endpoints to give you a better delineation of risk. Now with the European systems, most of these will divide our patients into 3 or 4 categories of risk. And sometimes with these lower levels of risk, we have some trouble predicting with intermediate risk. The US systems are primarily the REVEAL scores, and we have the REVEAL 2.0 score, which is the parent score, which summates a number of important variables that we glean in our clinical trials to give you an absolute score. And we have an abbreviated score of this called REVEAL Lite that Marius referred to also, which reflects only modifiable risk factors. And with the natriuretic peptides reflects both BNP and NT-proBNP. Now, the REVEAL score score, different than the European scores, it's able to give you either 3, which are the categorical risks that we've seen most often in our studies, but also can give you 8 full lines of risk, which allows a better delineation of the intermediate and high risk scores. And this is something that we'll go over a little bit later. Now, how can these scores meet the caveats that I showed you in the beginning? Well, risk scores can predict outcome, and they can predict both survival and in some cases clinical worsening, both very important for what we want to prove with our clinical trials. And this is just an example of the survival curves that you can get with the full reveal score that shows you a very nice delineation and predictability of mortality over 1 year or 5 years, and can also, very importantly, give you estimates of clinical worsening. And this is typically shown here at 3 years, but you can see that it gives you a very nice depiction of clinical risk. And if you even look at the low risk factors, you can see that you can segregate these to such a fine point that you might be able to find patients who are still ultra low risk where monotherapy could be appreciated. So risk scores do correlate with survival, one of the important first characteristics. Well, risk scores can also measure functionality. So we showed you how they mention survival, but we also want to make sure they correlate with how a person functions. And when you look at the individual scores, you can see that components of these scores, as mentioned earlier, are reflections of how a patient functions, both using the NYHA class and a 6-minute walk test, as you can see here for both reveal calculators, and also in our European systems. Also, you can see that both functional class and 6-minute walk test are important components. So how a patient survives and functions are incorporated into the contemporary risk scores that we have right now. But risk scores also have to correlate with how a person feels. And this is where the correlation between quality of life is very essential. And you can see also that risk scores do a very nice job correlating with how a person feels. And in this case, looking at emphasis 10, one of the quality of life scores that we use in PH, you can see how each of the European and the US scores correlate very nicely with this. So I think I've shown you now that a risk factor can correlate with the basic caveats, how a person survives, functions, and feels. Well, let's go a little bit more in depth about this. Well, can risk scores meet the caveat of a drug, score, and outcome interactions? Remember, this is particularly important to what the FDA and other regulatory agencies want. Now, if you look in this slide, which is a presentation we recently gave at CHEST, looking at changes in reveal light 2 categories over time in the Griffin trial, you can see that that drugs do change scores over time, and they do this with a high degree of fidelity. And this is nicely exhibited in this histogram. We can also see that now scores are also associated with change in outcome, whether it's between an active drug or placebo, as shown here, or within the individual outcomes for the drug itself. So we see that those who become low risk have the best survival, and those intermediate risk with the improvement a little bit less than low risk, but obviously a great difference between low risk and high risk. So now we've shown that a score that a new drug can change a score, and that these changes in score are predictive of subsequent outcome. The score systems that we have really met all the caveats set forth for a good clinical endpoint. Well, and as I mentioned earlier, it's really important to make sure that risk scores are validated. We have to make sure that these scoring systems are solid before they can make determinations of drugs that are being used to improve survival in our patients. And as we can see here, both the reveal scores have been validated retrospectively in a number of different subsets and registries and clinical trials in a very large harmonized FDA data set with very good C indices denoting high levels of discrimination. And similarly, the European systems have also been validated retrospectively in similar subsets. The other things we need to know about risk scores before we make a decision to use them as a utility and endpoint, I think are important to discuss also. And we'll go through some of those points. It's very important that risk scores differ in discrimination. So the predictability of each score, which is determined by its discrimination index, is very, very important to consider before using these in our clinical trials, because we want to make sure that we have the scores with the highest discrimination that can accurately predict the outcomes that we want them to predict. And when we look at a statistically sound tool, they have to be derived from sizable cohorts. We know this. In addition, the industry standard for applying a risk score at the patient level has to have a discrimination at least greater than or equal to 0.7. And this is shown in many of the common risk tools that we use in our clinical environment. And some of these are shown here in this animation, keeping the C index greater than 0.7. So where do we stand in far of the scores that we have for pulmonary hypertension? And I'm sorry, the animation is just a little slow. When we look at the different scoring systems that I demonstrated, again, you can see that depending on the system that you use, whether you use the Compare 1.0 or the French invasive or the new Compare 2.0, you usually can get 3 to 4 lines of risk. With Reveal, you get 8 lines of risk. And I think the discrimination shows that the more lines of risk, the better it is from a discrimination standpoint, especially when you look at the varied C indices that you have on these various scores. The other thing we need to know about risk scores is that risk scores tend to improve and from a discrimination when you use the full spectrum of the score. And this can be shown very nicely here when you look at Reveal 2.0, whether you use a 3-category system or the more 8-category system, and similar things you see with the abbreviated systems too. Now, risk scores predictive abilities increase with reassessment. That's an important caveat to know. And you can see here in the recent study that Marius told us about that when you use Compare 2.0, when you reassess these scores, the discrimination always increases better. And that's something that we need to keep in mind. These tools are very easy to use. And no matter if you use Reveal, Reveal Lite, the French, these really things can give you an accurate score within 60 seconds. So they're very, very easy to use in clinical trials. Risk scores can also facilitate how we devise our clinical trials and what we conclude from our clinical trials. This is an example in which we use risk scores to enrich for clinical trials. And this is looking at Ambition, Griffin, and Seraphin. And if we had enriched these patients using risk scores for intermediate or high risk patients, you can see that the sample size reduction would have been anywhere from 15 to 45%. We would have saved a lot of time doing these trials if we had enriched them for the patients that we knew were going to have the outcome. And this translates into improved economics for the study. And you can see when we enrich using risk scores, we can actually save about 40% that we typically spend on a clinical trial. Risk scores can also give us more information about what we want and what we're trying to prove in a clinical trial. In the example of the Ambition trial, when we stratified people just by NYHA class, we saw that combination therapy was better than pooled monotherapy. But if we would have stratified this trial according to risk, we may have seen something a little bit different. And that combination trial was much more of a beneficial effect in patients if they're enriched for intermediate or high risk as compared if they were low risk patients. So higher discrimination matters for making important therapeutic decisions and making conclusions about our clinical trials. Risk scores can also be used now as an endpoint in the era of COVID because many of these things can be done virtually. We can assess NYHA class. We can use telemonitors to assess vitals. So these scores can be used as part of a telehealth approach in our clinical trials going forward if the pandemic continues to unravel as it does. But with that, I'd like to conclude and thank you for your observations during this, and be happy to answer any questions during the panel discussions.
Okay, thanks a lot, Ray, for the nice presentation. Now the next speaker is Joanna Preston from Tufts University in Boston, and proposal of a novel endpoint based on actigraphy.
Hello, everyone, and thank you for having me today. Amazing conference, amazing 2 sessions. My task is to talk about proposal of novel endpoints based on actigraphy. And everyone on the panel and in the audience knows that I'm a very strong proponent of actigraphy. However, I will try to be unbiased. So we know that the assessment of physical activity can be quantified by, measuring granular activity with any of the devices that are available nowadays, and there are several hundreds of devices, and the activity intensity can be quantified into sedentary, light, moderate, vigorous activity, and this pie chart and table summarizes the types of activities that are fitted into these 4 categories. However, in the more significantly impaired patients, the sedentary and the light activity can be even broken down into 3 more categories. Now, every Dick, Tom, and Harry has a device. As John Owen in the 1600s used for the first time this quote in an Oxford talk, and it's stuck to us even in the US and worldwide. So we all have smart devices, smart watches, smart activity, wrist devices or ankle devices, or even some of folks have tattooed devices that measure their activity. So it's a worldwide. So why should not we use this information to understand how patients do in clinical practice, but also how new drugs that we're developing perform in everyday activity, which is a more patient-centric way to assess how patient well-being is. A tool that the FDA loves to look at nowadays. So, this is a table that summarizes crudely the activity devices that were used in clinical trials in different cardiopulmonary disorders. However, these devices have been used widely in other clinical trials, such as healthy volunteers, athletes, depression, renal disease, and so on. So they have been used increasingly in clinical trials in all sorts of cardiopulmonary and non-cardiopulmonary conditions. So they are here to stay. It's just that we have to become smart to know how to use them in our pulmonary hypertension patients. In heart failure patients, it's not only heart failure with reduced EF, but also heart failure with preserved EF that these devices have been used, and not only by pharma, but also by the NIH clinical trials. So if you look at the few reports in pulmonary hypertension using these devices, there's definitely a comparison between the 6-minute walk distance and the step count in pulmonary hypertension patients. Patients with prevalent PAH showed that the 6-minute walk distance was different between functional class 1, 2 versus 3, 4, and that was paralleled by the daily step count in these patients. Now, however, very, very small group of patients. But not only in adults, it's also in PAH children that these accelerometry devices have been studied, which showed that in a small group of PAH children, the moderate physical activity, vigorous physical activity, and the combined MVPA, which is moderate and vigorous physical activity, was associated with long-term outcome. Again, small studies, but that suggests that this type of long-term granular activity monitoring device can be informative of the well-being and long-term outcome of our PAH patients. Now, the only randomized clinical trial that used an accelerometry device is TRACE in PAH patients to test activity using the Actigraph device. And this table summarizes the results of this trial that enrolled largely very stable PAH patients who were the vast majority on double oral therapy and were randomized to either selexipag versus placebo. And I think this trial, although not statistically significant in the main actigraphy counts, which was daily steps per awake time, daily steps in non-sedentary activity. It was not different between the selexipag group versus placebo. It sets the ground to measure what is the baseline activity of our patients, and that is very informative in our hopefully future clinical trials if we are to use this kind of measurements. So there is a relationship between physical activity, walk test, and heart and quality of life in PAH patients. And this table highlights that there are several parameters that can be taken into account when we design future clinical trials, such as number of steps a day, which has been associated with function, physical function, general and mostly vitality, health, as well as mental health, social function, and so on. And the sedentary, moderate, and vigorous activity breakdown shows that the more active these patients are, the better they feel within this type of questionnaire. So there seems to be a relationship between patients with PAH moving more and feeling better holistically. Again, these are very small studies. Now, before I wrap up, because this talk had to be really short, more generative of discussion, let me be a debater on my own and bring on a couple of heart failure clinical trials that bring up certain questions of how to place this type of functional activity measurement within our future clinical trials. So in awake heart failure, and I welcome the input of our cardiology folks because they are much more previewed on the heart failure clinical trials than I am as a mere pulmonologist. So the awake heart failure clinical trial enrolled a very small subpopulation of 140 patients only who were randomly assigned to the combination therapy of sacubitril and valsartan versus enalapril for 8 weeks. And then it was followed by 8-week open label trial. Now, we do know that this combination therapy was associated with long-term survival outcomes and risk of hospitalization. So it was reasonable to look to see if this combination in a subclinical trial made an impact on the physical activity and functional activity of patients with heart failure with reduced EF. Now, look at the primary endpoint. It was the change from baseline in mean activity counts during the most active 30 minutes per day at week 8. And it was a geometric measurement of this change. Now, the reason for the main outcome of this, most activity of during the 30 minutes per day was chosen because there were certain clinical trials that show that this particular measurement has been associated with a good outcome. So if you look at the graphs, the red bar is are the patients who are on a combination therapy. The black bar is the group assigned to enalapril, and there's absolutely no change. So even though the long-term significant outcome endpoint has been significantly better with a combination therapy, the functional activity measured by an actigraphy type of measurement with this particular count was not different. And that brings me to the question to you guys: what would be the best measurement given by a long-term, 24/7 actigraphy type of activity device, or an activity device that would be important and significant in our PH patients? And of course, I don't have to remind you, our PH patients die of heart failure, albeit not by ventricular failure, but by right ventricular failure. So is there can we translate what we learned from the heart failure trials to the RV failure trials and PH trials? So that's another question. The other question is let's look at the outstep heart failure trial. So this trial enrolled patients with the same two treatments, enalapril in the blue and the combination therapy in red, which again showed long-term outcome improvement. And on the left-hand side, it shows the improvement in 6-minute walk distance, which was not statistically significant between the two arms, but it was positive. And on the left-hand side, the B graph shows the activity measured by an accelerometer device called Motion Watch after 12 weeks of the treatment. And if you see, the activity of these patients in both groups went down. There was no difference between the groups. However, the highlight is the discordance between the 6-minute walk distance measurements and the activity device. Now, I have to remind you that in heart failure studies, there are a couple that show that a mere 10-minute increase in physical activity or a 5% increase in exercise capacity was associated with beneficial effects on mortality and risk of hospitalization. So how can we reconcile these information that are so significant because they are in a larger group of patients with heart failure, and can they be translated into our PAH? Is really an activity day monitoring way really reflective of the cardiopulmonary capacity of these patients because of an improvement in a drug treatment? Can these patients be that they can do better on the 6-minute walk test, which is quote unquote unencouraged? However, my patients come with a tennis shoes and then they're ready to do the 6-minute walk test. It's not encouraged, obviously, but maybe subconsciously it is, versus their everyday activity where they're usually sedentary, and if they're not pushed, they will not do, even they can do more. So even though I'm a huge proponent of accelerometry in the future clinical trials, I'm not sure if it really reflects the cardiopulmonary capacity to do better with a new treatment unless you incentivize these patients to do more. So instead of conclusions, I have questions for you. What measure?
Rapidly, because it's very late.
And then these are the questions, and I will leave it up to discussion and look forward to your input. Thank you so much.
Thanks, Ioana. That was great. You know, my Apple Watch is screaming at me right now, as you can imagine. In the interest of time, let's move on to talk about another really important potential endpoint, and that is patient-reported outcomes in PAH. I would like to welcome our colleague Simon Gibbs from London to give that talk. Simon?
Well, thank you very much, Val. Good evening to everybody from London. Good morning, good afternoon, good evening. It is easy to put aside the emotional impact of disease in clinical trials, especially in something like PAH, where we've got very good pathophysiological endpoints, which play such a central role. I am planning in the next few minutes to say something to you about when and how we might use patient-reported outcomes, and also quite a lot about the huge challenges. And I'm going to just choose, anecdotally, what I think are interesting aspects of this that we need to think about, because it's a subject that could last all day if we really wanted it to. Here are my declarations of interest. And I want to start with the case for a PRO, and that is really pretty simple. The impact of pulmonary hypertension on patients' overall quality of life is massive. And in addition to that, there's a very significant number, an enormous number of patients with problems with emotional well-being. And when we asked these patients in this British study, what is it you hope to gain most from your treatment of pulmonary hypertension? The answer was they wanted improvement in quality of life and increased life expectancy. This, I put to you, is the case for considering a PRO. Now, the traditional endpoints which we have in PAH measure pathophysiological things, but they do not tell us about how the patient feels. To know how the patient feels, we have to measure quality of life and make the link between these two so they can be joined together. And then we have to communicate to patients what the results of the quality of life are. The health-related quality of life is part of patient-reported outcomes. And here is the FDA and the EMA definitions of PROs. I would just note out of interest that when these PROs talk about the status of a patient's health condition or the patient's perception of a disease, there's no direct mention of the emotional well-being of the patient. Now, the first step is to create the PRO. And this process is important because we have to get it right in clinical trials. And there is no correct way of creating a PRO. This is the FDA's proposed template, which is really an excellent starting place. It has only been used, to my knowledge at least, explicitly for one PRO, which is PAH SYMPACT, which is being used in PAH trials, of course, as I speak. Remember that in a trial, you need to think very carefully about what questions that are relevant to the target product that is being investigated in the trial and that is relevant to the aims of the trial. So it isn't a one PRO fits all. And we should bear that in mind. It may fit many, but it doesn't have to fit all. Now, when it comes to using these PROs, there are very many challenges and limitations. There are enormous lists if you want to go and look for them. But I want to put to you the issue of choosing the right questions, because I think a lot of the time we talk about PROs, but we don't actually look at the questions ourselves. And when you look at them, you see how different they are in different questionnaires and why that might be important. In this slide, you see on the left, PAH SYMPACT. This is half of PAH SYMPACT, the symptoms, and on the right, emphasis 10, which is 10 questions. Now, PAH SYMPACT is a multi-dimensional questionnaire. Emphasis 10 is a unidimensional questionnaire, which is only related to health, to quality of life. PAH SYMPACT is symptoms and impacts. And I've drawn some lines here between the things that might be related, and you'll see they are completely different. And indeed, if I go and show you the impacts now, then similarly, they are different, and one is asking different questions. There are many other challenges. I just want to raise this one because it was raised with me by a patient, and that is the issue of the quality of life. So that is the quality of life from a trial. And the patient said, well, yes, but I'm a young mother, working part-time, I've got young children, and why would the things that affect my quality of life be the same as those if the population were retirees? And I think these are a lot of the points and nuances that become important in a clinical trial. The World Symposium has made a number of recommendations over the years. The first is to include PROs as a secondary endpoint in clinical drug studies, and this is almost universally done now. Including patients in the clinical design process, I think, is very important if you want to get overall well-being into the trial. And using PROs as a clinical trial outcome, I'm going to come back to in just a moment. So where is all this leading? I would put it to you, we need to involve patients if we're going to use PROs, which I think I've made the case we should, because it's what patients want. We're going to use them. We need the patient in the clinical trial steering committee not only to be involved in how does it fit in with the endpoint plan, what are the questions that are relevant to this medicinal project and this particular population, and how are we going to communicate the results to the patients at the end of the study. That is why you need to have help with publication. The PROs are frequently, when they're measured in trials, they're secondary endpoints, they get reported, of course, in the paper. But the patients, I wonder how many of your patients actually know the PRO results of the drugs they are taking from their clinical trials. I'm embarrassed to say, I don't think I've ever talked to a patient about this, but we need to. And then the next question that is important is, how do PROs rank, in your opinion, in clinical trials? Well, as Ray Benzer has pointed out, PROs can be related to other factors, and here is how the emphasis 10 score relates to survival in 1,700 patients in the UK. So they're not simply standalone, they do have relations to other things that are being measured. Now, I've taken, for the sake of argument, a traditional composite endpoint, and this is an endpoint that has been used before, of course, in PAH trials. But I put it to you, with the patients talking about quality of life being so important to them, why are we not able to include patient-reported outcomes in the composite endpoint of clinical trials? I do not pretend for a second it's just a matter of dumping in it and doing it. This needs to be investigated, it needs to be validated well before we actually get involved in doing it. But is this not the aim? I put it to you. So, in summary, patients with PAH are seriously affected, their emotional well-being is seriously affected by the disease. PROs are a powerful way of telling us how patients feel. We don't have any other way of doing it. Patients with PAH should be involved in clinical trial steering committees because this is how we get PROs done properly in trials, and they need to be tested as a primary endpoint, not automatically put in, but tested. Thank you for your attention.
Thanks a lot, Simon. Now we move to the last speaker of this first session, Rebenza again. The talk is on hemodynamic remote monitoring endpoint. Please, to respect the time, because we are very late.
Thanks, and for round two, and again, I apologize to the moderators. I'll try to keep a little bit better on time with this second talk. Being Sicilian, we tend to be a little long-winded, but I will try to counter that in this talk. So the purpose of this next talk is to try to really introduce a new way of managing and monitoring our patients using remote hemodynamic monitoring, and whether this might be a good endpoint for future clinical trials in pulmonary hypertension. Now, as we all know, that we've kind of moved away from hemodynamics, particularly with our contemporary risk scoring systems, and have really tried to focus on non-invasive ways to predict outcome in our patients. But I believe hemodynamics continue to be a very, very important means to predict outcome of our patients. And you can see this beautifully demonstrated in a variety of different studies, both in Europe and in the United States, where hemodynamic factors continue to be an extremely important part of our armamentarium to predict outcome. Particularly, they continue to be incorporated in more sophisticated scoring system using Bayesian analysis. So hemodynamics important, and why are they important? Well, if you look at the progression of decompensation in pulmonary hypertension, you can see where the parameters that we typically monitor in our patients, including risk scores, and their relationship to the onset of right ventricular failure and hospitalization. And you can see that many of these are very late changes in this paradigm. But when we look at hemodynamics, you can see that these really give us an upfront advantage in determining eventual outcome of our patients, and using hemodynamics give us a huge gain in time to predict outcome and to reverse processes that may lead to deleterious effects in our patients. So hemodynamics are important. Now, the problem with them is they're gleaned typically through invasive studies, and we all know the shortcomings of right heart catheterizations in our patients. So there are better ways of doing this that omit these disadvantages of the invasiveness, the risk, the single time point estimates, and the answer is yes. We now have new devices like the CardioMEMS device that give us real-time information on hemodynamics daily in our patients, and can be used tentatively as an endpoint in clinical trials. And this is the typical information that is gleaned from these hemodynamic devices. As you can see here in this chart, you get the mean pulmonary artery pressure in blue, the pulmonary artery systolic pressure in red, and the pulmonary diastolic pressure in green. And you can also get an area under the curve of pressures over time that you can monitor and are tightly correlated with changes in therapeutic management. And the CardioMEMS has been validated in a very large clinical trial in heart failure. And you can see that those patients who were managed with this device had a much lower risk of hospitalizations when patients were managed actively and in real time using these hemodynamic measurements. And this device has now been approved for the use of management in heart failure. Well, in order to use the device, we have to be confident that patients will use it and that physicians will use it. And when you look at the post-approval trial of the CardioMEMS device, you can see that compliance among patients in giving you these transmissions is very good over time, and that physicians continue to use these pressures to manage their patients. So good compliance, both from the patient and the provider standpoint, indicates that this device can be very useful in the real-world environment. Well, safety is something that we also have to consider when we want to use these in clinical trials. And you can see the long-term safety for these devices are pretty good, with a 99% freedom from device or system-related complications that adhere. And also very important, these sensors are good and they last long. And you can see on the right-hand panel that the freedom from sensor failures is also very good with these devices. Well, I think many of us realize that just looking at pressures as an endpoint in pulmonary hypertension may be somewhat limited, but we're not limited thus far now in using these new devices because we've also been able to validate a means to determine stroke volume with these devices. And stroke volume gives us the opportunity to measure a whole variety of other clinical parameters that we feel are important in determining outcome in our patients with pulmonary hypertension. And you can see the correlation between the cardiac outputs and stroke volumes gleaned from this device are actually pretty good when you compare them to right heart catheterization. So in essence, these measurements are very dependable. So now we have a device that can give us pressures and estimates of stroke volume and derivations of hemodynamics using stroke volume real time in our patients on a day-to-day basis, which could be very, very helpful in managing a clinical trial. Well, is this device safe and feasible to use in pulmonary hypertension? We conducted a small study looking at this device in patients with very severe pulmonary arterial hypertension, as shown on this slide. And what we saw that was this device was very helpful in gleaning varieties of hemodynamics over time. And you can see here the changes in mean pulmonary artery pressure, stroke volume, total pulmonary resistance, and compliance using this device long-term in this short and small trial. You can also get an essence of the changes in pressures using different sorts of management. And in this small trial, again, you can see the differences in hemodynamic changes in someone who is highly managed or moderately managed with therapeutics. Now, these hemodynamics seem to correlate well with standard things that we feel are important in measuring outcome. And in this instance, correlating these hemodynamics with 6-minute walk test, again, in this small trial, showed good correlation with a standard endpoint. Well, these devices can be used very nicely to manage patients. And just to orientate you to this slide, in the upper panel, you see the 3 primary pressures. And then the hatched lines are an investigator titrating intravenous prostacyclin from a slow titration to a more rapid titration. And at the bottom panels, you can see the changes in other hemodynamics, including heart rate, stroke volume, cardiac output on the left, and then stroke volume index, total pulmonary resistance, RV power, stroke work, and compliance on the right. And you can see that you can very nicely delineate the changes in these hemodynamics using these devices with changes in therapy. You can also use these devices to transition patients. I know we talked about this early in one of the panel sessions, but these devices give you a unique opportunity to determine if changes of hemodynamics are moving appropriately with transitions, and if not, you can save these patients by putting them back on the management schemes that seem to do the best for them. And that's the advantage of using this, is because these changes in hemodynamics occur way before symptoms can occur. You can also follow these patients and actually make predictions about end of life. And this is a patient who failed therapy with pulmonary hypertension. And again, you can see the detrimental changes in cardiac output and stroke volume related indices using this device very nicely real time. This device can also be very nicely correlated with other standard images to determine RV function, which we know is an important determinant of survival in these patients. So hemodynamic changes correlate with actual changes in RV structural and function long time using these devices. These devices can also be used during a 6-minute walk test to note change in pressures with exercise. And I think this is a very important piece that we've yet to glean, or is more difficult to glean using standard right heart catheterization. And as you can see here, you can see the changes in heart rate and mean pulmonary artery pressure pre-6-minute walk test and post-6-minute walk test, and how these changes are modified over time using different therapies. And you can see also in the more dynamic hemodynamic principles that we can check, like stroke volume, cardiac output. And you can also see that very interestingly, that the changes in cardiac output that occur with the 6-minute walk test in our patients may not be necessarily correlated with changes in stroke volume, but more related to changes in heart rate. So this is kind of the interesting information that you can get with these devices. I think because of the success in heart failure and then the limited experience we've had with pulmonary hypertension, we've now been able to work with the FDA and several pharmaceutical companies to use the CardioMEMS device as potential endpoints in clinical trials. And these are example of 2 trials: the Artesian trial, which is going to be a phase 4 trial using the CardioMEMS, which is going to be employed to titrate parenteral prostacyclins to a goal mean pulmonary artery pressure, and in the phase 2 Cereno trial, in which we will explore 3 doses of CS1 using the mean pulmonary artery pressure to determine the doses to use in the next pivotal placebo-controlled trial. So in conclusion, I think the implantation of these devices are feasible and safe in heart failure and pulmonary hypertension. Compliance is good among patients. It's clinically useful to predict events, and also can be used as a virtual endpoint in clinical trials in the era of COVID. And with that, in 16 seconds, I'll give it back to Dr. McLaughlin and Dr. Cereno.
Great. Thanks so much, Dr. Benza. That was really a tour de force of the CardioMEMS device. We are now going to move on to the second section of our program, and it's really going to be viewpoints from various stakeholders. And we're going to first start with the investigator viewpoint, and we have 2 wonderful investigators, Thenapin from Minneapolis and Rohan Zamanian from Stanford.
Well, thank you very much, Dr. McLaughlin and Dr. Cereno. On behalf of Dr. Thenapin, I'm Rohan Zamanian. We're from Stanford University and University of Minnesota. Our portion is going to be rather short. We'll stick with the time here you provided, and we're providing the input from an investigator's perspective. And I guess we had discussed this, and you know, we're sitting here on a virtual platform because of the global pandemic that does not seem to abate. So our thoughts are initially to put into conversation here is a view towards being pragmatic. Global pandemics are rare, but it has shown us the capacity of how regional, national, or global disasters would interfere with the successful conduct of clinical trials and the care of our patients with pulmonary hypertension. We can't tell you how many times patients did not have the opportunity to come to clinic to conduct their 6-minute walk test. And so as we begin to look to the future, as we begin to talk about the environment of innovation here, a lot of the conversations, as Dr. Preston, Dr. Benza, and others have mentioned, is about innovating clinical trial endpoints that could be brought into the home and consider the patient's perspective on how they feel, function, and survive. Key words that we've heard a lot about today. So from an investigator perspective, as a stakeholder, we would want pragmatic ideas to be brought forward into future clinical trials. You've seen the data around actigraphy. My good friend and colleague, Dr. Kaywood, has just published data on accelerometry data in the Phantom clinical trial, linking the relationship, as has been mentioned, between accelerometry, 6-minute walk distance, and quality of life. We would love to see more of these measures brought forward into early and late phase clinical trials and incorporated, as Dr. Gibbs has mentioned, into endpoints that are really formally placed within the clinical trial environment themselves. So a look towards pragmatic ideas, and it doesn't take just a global disaster to bring that home to us. The second conversation that comes up here a lot from the investigator's perspective is the concept of precision phenotyping. As we've already discussed, the success in the last 2 decades of therapeutics has provided a better symptomatic management and outcome of patients with pulmonary hypertension, but pulmonary arterial hypertension, but really no cure or a disease modification has been proven yet. There's early days of that, and there are some exciting results from the sotatercept studies that are coming forward. But it's become more challenging, is obvious from an investigator's perspective, that to demonstrate treatment response is something that is a challenge for the next decade of clinical trials. So from investigators' perspective, we would love to see the concepts of precision clinical molecular phenotypes and how they empower and define the concept of disease modification. As Dr. Cereno pointed out, it's very difficult to show impact of therapies on hard outcomes like survival. It's a rare event in PAH. But what do we think as a community? What is this concept of disease modification, and how could we prescribe a definition to this term using the technologies that we're talking about, imaging technologies that are being utilized currently, like the vascular imaging work that's being done in several clinical trials with our colleagues at Fluida, molecular signatures, and then the future of artificial intelligence, machine learning, and those advanced analytics and how they would apply to this precision phenotyping process. And really, the last thing is a view towards what we call raising the bar. And what I'd like to do, maybe Thenipan, I would love to pass this on to you to chat a little bit about it, and we go from there.
Sure. Thank you, Roham. And first of all, I want to thank the organizers and our chair, Dr. McLaughlin and Dr. Cereno, for giving us this opportunity. So continuing with Dr. Zamanian's viewpoints from investigators. So the next viewpoint from an investigator perspective, we would like to raise is, why not we raise the bar high now? Thanks to everyone in this call and to everyone working in this field, we have made tremendous progress in improvising the endpoints for PH clinical trials in the last 3 decades. Very nicely discussed this morning. We started from 6-minute walk distance to time to clinical worsening, but perhaps it is time to raise the bar high. And Professor Cereno very nicely, elegantly explained why survival cannot be an endpoint in PAH trial, but maybe now this could, with the tools we have, we should or perhaps consider survival alone as a primary endpoint in future clinical trials using the tools we have. We can do enrichment clinical trials in high-risk category patients, perhaps use the reveal risk score or any risk score, take a high-risk patient where the mortality rate is very high. We still have a very high mortality rate, and use an adaptive trial design to reduce the number of patients. So the limitation of using survival alone as an endpoint is it's rare and we need more patients, but why can't we use the tool, or should we use the tool we have to go back to the very hard endpoint of survival? And then in the interim, when we are doing that, from an investigator perspective, we would like to strengthen the definition of the composite endpoints. And we should try to, or perhaps use only hard endpoints, such as initiation of parenteral prostacyclin, heart failure, hospitalization, death, or lung transplant, and move away from change in 6-minute walk distance, etc. Because Dr. Halper nicely showed that the change in 6-minute walk distance or NT-proBNP actually per se don't translate into survival benefit. I think that's all. That's a review from an investigator perspective, and thank you for the opportunity.
Yeah, thank you, Val and Gerald. We'll give you back 2 minutes, 2.5 minutes.
Yes.
Thank you.
now we have to move to the viewpoint of industry, and we have three speakers with 5-minute presentation for each one. The first speaker is Bjorn Daloff from Cereno. The second speaker is Mahesh Patel from Merck. And the third is Philip Adamson from Abbott.
Okay, good day and good evening and everything in between. It's a great pleasure for me to be here today, and I really want to thank you, the organizers and the chairman, Dr. Simino and Dr. McLaughlin, for inviting me to this session. I'm going to give the small company's perspective on doing a phase 2 trial, and here is the highlight of the trial. It's our candidate CS1 in PAH. We have a primary objective of safety and tolerability, but we have also other objectives which are exploratory. And we have most of the standard efficacy endpoints for this patient group. We have validated risk scores. That was mentioned by Dr. Benza, pharmacokinetics and dose finding. And we use the CardioMEMS. We will do the study in US at 6+ centers, 30 patients. We hope to enroll the first patient soon and have top-line results in Q4 next year. So what is CS1? CS1 is a reformulated VPA, a patented formulation. It's a HDAC inhibitor with epigenetic modulation. It has a 4-fold efficacy shown from animal studies, both pressure reduction in the pulmonary circulation, anti-inflammatory, antifibrotic, and antithrombotic through a fibrinolytic mechanism. When we designed the trial, we use criteria that was published a couple of years ago. We look both at safety and tolerability, PK sampling. We look at mechanistic markers. We have a homogeneous population. We have risk tools as an enrichment strategy. We use change in risk marker of efficacy, and we have also saved biomarkers for future exploratory. The study population is a well-defined PAH population with a functional class of 2 and 3 and a revealed risk score of 6 to 10. As I said, primary objective is safety and tolerability. We will implant the CardioMEMS and have a baseline period to have the patient familiarized with the equipment and also be able to have a baseline measurements. We have 3 dose groups, randomized, 10 in each. It will be 12 weeks follow-up on active therapy, and we will have after that a follow-up of 2 weeks. So as I said, we measure a lot of endpoints in this. Most of them you are well familiarized with. It's both patient outcome reporting, and it's all kinds of functionality and structural changes, as well as morbidity mortality. We use actigraphy. We use the risk scores. We use the CardioMEMS. And some of them we have a blinded reading for. So this is the setup. And as mentioned by Dr. Benza, I will not go into the risk score, but it's well validated, and it has, I think, a true value and could be a primary endpoint in the future. So we will validate that. We will also have the CardioMEMS, and I will not go into detail about the CardioMEMS since it was elegantly presented by Dr. Benza, but it will give us a huge opportunity to evaluate our therapy. As I said, in summary and conclusion, we are a small biotech. We want to have as a cost-effective study as possible, that most information for the money. We use 3 doses in 30 patients. We use the CardioMEMS and do this study in collaboration with Abbott. We use validated risk score, and we also have measurement of movements like actigraphy. And this will be the basis for a larger study in the future, proof of concept study, placebo-controlled and double-blind. I'll give you 1 second back. Thank you.
Well, thank you for the kind invitation to this informative session. I'm an Abbott employee by means of disclosures and a stockholder of the company as well. I have no slides, but as a newcomer, I'd like to maybe contribute brief comments and some hopefully provocative speculation. So as a newcomer, I'm going to go way back in time, back to May of 1883, when Lord Kelvin suggested in a lecture that if we can't measure it, we can't improve it. And then a little bit closer to today, 25 years ago, we began developing implantable hemodynamic monitoring technology, primarily focusing on patients with heart failure, regardless of ejection fraction. We discovered many things over those years, but maybe the most applicable to this discussion is that well over 80% of patients with symptomatic left heart failure also have significant secondary pulmonary hypertension, much higher prevalence than we thought, and even less ill patients. Additionally, Professor Benza discovered that the PA pressure values we obtain in the right heart catheterization in the cath lab many times are lower than those observed in ambulatory patients. And in a series of patients with heart failure, Dr. Benza found that the mean PA pressure from the cath lab missed the diagnosis of pulmonary hypertension, in this case, secondary pulmonary hypertension, about 25% of the time. All of this led to the concept that daily assessment of PA pressures with the CardioMEMS heart failure system in particular, not only is superior to managing volume and avoiding decompensation in heart failure patients, but maybe a superior means to understand when drug therapy impacts vascular resistance and possibly right ventricular function. And I send a big thank you to Dr. Benza as well for outlining the first experience of using implantable hemodynamic monitoring in patients with WHO Group 1 PAH in his excellent presentation. You know, this is very exciting potential and may represent sort of a sphygmomanometer for the pulmonary artery, which may provide a means to develop interventions that safely and directly impact PA hemodynamics, similar to the therapy development for systemic hypertension. Hemodynamic monitoring has now been evaluated in well over 8,000 people with left ventricular failure, both with HFpEF and HFrEF, demonstrating very consistent safety and efficacy outcomes, and is now active in nearly 30,000 heart failure patients in the Western world. So now we move forward with investigating the value of this approach in patients with PAH. So as we move forward, there are several guiding principles that we've learned over the years and maybe could be considered as we move into novel endpoints and designs of clinical trials in this arena. First of all, there are several sensor concepts that are being developed, which include implantable intravascular devices like CardioMEMS, subcutaneous devices like implantable loop recorders, interstitial devices like Freestyle Libre with alternative analytes, and wearable devices. Combining these technologies may actually answer several questions and provide better gold standards for therapy intervention. We also encourage creative evidence development methods, which are needed, such as linking device informatics with large real-world databases that can give rise to conclusions from large patient populations. But we must remember, as Dr. Gibbs mentioned, to include the largest group with vested interest in the clinical trial and the actual workflow process and the outcome, which is the patient. We're in the MDIC program about to publish a paper with novel statistical methods to incorporate patient preference for risk-taking in clinical trial design endpoint selection and probability of effectiveness should also have patient input in the future. There's several benefits when we involve the patient in the process, including the engagement and adherence and in clinical trial practice, making the patient a partner rather than a passive participant. This enhances a sense of security. It's an amazing educational opportunity, and maybe most important, improves the locus of control in a chronic disease that many times takes that away from them. So I submit speculatively that direct frequent PA pressure measurements in this population may make vasoactive compound development much more efficient and provide more direct endpoints in the context of safety evaluations. Thank you again for the invitation. I look forward to the discussion.
Hello, everyone. My name is Mahesh Patel, and it's a pleasure to be with you here today. And thank you, Dr. McLaughlin and Dr. Cereno, for the opportunity to speak. So I've learned a lot from the discussion today, and I think what's very interesting from the discussion is how, despite the limitations with some of our conventionally used endpoints in phase 3 PH trials, like 6-minute walk distance and time to clinical worsening, despite their limitations, they have led to very important impacts into the care of PAH. And so as we think about new endpoints to introduce into phase 3 programs, we must think really critically about how they will advance from the already successful paradigm that we have. So if you could advance 2 slides. Thank you. So what I'll provide here are some of the key considerations that I, as a clinical trialist and cardiologist at Merck, think about as we're selecting endpoints for phase 3 trials in PAH. So first, and this has been discussed a lot, are all the stakeholders. And of course, there are many, and starting number 1 at patients, but of course, caretakers, clinicians, regulators, and payers. And we are listening very carefully to all of these stakeholders as we think about the right endpoints and strategies we have for our phase 3 trials. One very important thing is that not everyone agrees, and I think that that's no surprise to this group. And so how do we deal with that level of disagreement? And I think predominantly our focus is not thinking about how to answer every question in a single phase 3 trial, but instead, how do we design a menu of phase 3 trials that can ultimately answer the broad questions and important questions of interest by all key stakeholders. The next thing, after discussing with stakeholders, we think very methodically about how to answer these questions in the most trial efficient way. So clearly we're looking for endpoints that are responsive to the products and mechanisms that we're looking to affect. Also, we're looking for endpoints that have low variability. But we go beyond that, especially for those things that are primary, secondary endpoints. We are looking for ways to reduce the variability. Perhaps, for instance, with pulmonary vascular resistance, we think critically about what type of core lab we should have. How should we standardize the assessments so that we can reduce variability? Also, it's critically important to us as we design these trials, our missingness is really kept at a minimal level so that these trials can truly be efficient. Now, amongst all of the endpoints that we're considering, at Merck, we are really interested in hard cardiovascular clinical outcomes. And so that brings to the third point here. And I've listed here as 2 key examples of that are PAH hospitalizations and mortality. And I think clearly this is a challenging endpoint across the board for any cardiovascular trial, but particularly so for a rare disease like PAH. So how do we think about designing trials here? And I think we have to be quite clever in order to do this successfully. And so first, we have to think that we need trials that have the right number of events to be appropriately powered to detecting these changes. So that is key. And that can be, that number of events can be increased by, first, having more patients in the trials, so larger sample sizes. Secondly, having longer durations, and longer durations not just on with patients in open label, but really placebo-controlled long trials. That's really what's needed to accumulate the large number of events that are going to be needed to discerning important differences on this key outcome. Next, we can think about how do we use enrichment criteria to select these patients that may more likely have an event with a reasonable amount of time. Now, there is a downside to that, and that is patient selection, and then we, always struggle then knowing how to apply this type of data and information into clinical practice, because what was studied was maybe just on this enriched population. So another way we're thinking about it and exploring this with regulators is, how do we think about combining different trials, pooling them in a way such that we can ask different questions in different trials, but then ultimately have a large enough data set. And these data sets have to be appropriately designed in a way to allow for that. But if we do it smartly, we may have a more powerful data set that would enable us to answering this important question. Next, we also obviously are thinking about feasibility, and this feasibility goes very much into not thinking about it as one trial, but in the range of trials that can really answer all the questions we have when we think about this pre and post approval. And then finally, we're looking for the endpoints that are validated and standardized such that we'll be able to compare the effect of our drug on these outcomes against different compounds. So thank you for your time, and I look forward to the remaining part of the discussion.
Great, and thank you for persevering through the technical difficulties there. We will now move on to the regulatory viewpoint, and we have two speakers, Krishna Prasad from the MHRA in Great Britain and Mitch Satka from the FDA in the US. Thank you.
Hear me, I'll start talking. This has been a fascinating discussion and then very good session for talking about multiple endpoints and various perspectives. There is very little to particularly say much about, having had the discussions about regulatory viewpoints from various other speakers. One of the things that struck me as the discussions went on was that what had been described in the regulatory guidelines and other regulatory requirements. And looking back at the European guidelines, suggests that this is the exercise capacity and the time to clinical worsening that included 4 subpoints, which is all-cause mortality, time to unplanned hospitalization, time to deterioration, and right-sided heart failure. Sequentially, many of the European guidelines have said that the hemodynamic measures were not sufficient and were not specifically patient-oriented or quality of life-oriented. So what does a meaningful endpoint could be for us? I think we should use clinically relevant patient data and outcomes. And I'm saying this because the patients are funded information, and we do need to incorporate what they require and what they offer to the clinicians and to the clinical trialists. Meaningful endpoint should transform a measurable parameter into a validated regulatory standard if possible. And in my mind, it should be broadly applicable to different forms of pulmonary hypertension in this particular context, not necessarily in the same fashion, but somewhat differently necessary. We should be able to use varying monitoring opportunities, whether it's home monitoring or continuous monitoring, and local data packages to facilitate both the regulatory approval and provision of this agent to the patients. whilst considering potential for public health impact and reimbursement. Many of the speakers have said what that would require, what it should be. And in the context of risk assessment tools, they've also discussed specifically sound risk assessment tools. And Dr. Pendler points out that they should be derived from sizably well-defined cohorts, and I accept that strongly. And it should be composed of risk parameters, and we should be evidence-based. It should be validated internally and externally. And lastly, I would expect that to have a high discriminative value. So, sort of in summary, what I would expect is that the clinical meaningful endpoints, and I'm sure I'm speaking for many of my colleagues, this should measure how a patient feels, functions. We discussed whether 6-minute walk test is a really valid endpoint, given that many of them did not differentiate, or the size of effect wasn't sufficient. However, from a patient's perspective, 6-minute walk test and daily functionality is a very critical event for them. I think we have probably not done the right service in maximizing that opportunity. The second question that I would like to focus on and summarize is that we should influence any of the endpoints that we have and we use through interventions or drugs. And we must finesse the ability to measure these impact therapies. And we should be able to accommodate variation in effect size. I recognize that there are very few of these, and so we need a concerted effort in identifying such endpoints. Thank you very much for the invitation. This has been very helpful, and I've learned a lot in the session.
Hi, my name is Mitchell Satka. I'm going to today represent the Division of Cardiology and Nephrology at FDA. And I just want to highlight a number of topics that have come up over the course of today's sessions in the study of pulmonary arterial hypertension. And I want to start just by encouraging that we as a division encourage the development of novel clinical trial endpoints for pulmonary arterial hypertension. And really, we recommend early and ongoing discussion with our review division and any others that might be involved if you would like to propose a novel clinical trial endpoint. To start, I think we have clearly embraced and are supportive of risk enrichment strategies and will often give a broader indication for therapy beyond the strict entry criteria for a clinical trial that perhaps uses those risk enrichment strategies. Regarding adaptive trial designs, I think that we agree that these may allow for more efficient clinical trials. They facilitate dose selection in phase 2 and reducing risk for an uninformative or underpowered clinical trial in phase 3, and thus do what is best for patients. Of course, pre-specification is critically important in these situations. I want to comment on the topic of Bayesian approaches. I think we typically utilize previously generated data or priors regarding a therapeutic to help interpret subsequent trials. And you know, for single trials, we require a very statistically persuasive statistical calculation or a low p-value. And for subsequent trials, we require slightly less small of a p-value. And I think in that way, Bayesian approaches kind of have an unclear utility in this context, in that you know, we may agree to utilize them, but sponsors may actually find that the Bayesian kind of goalposts require similar overall supportive evidence to a pre-negotiated frequentist approach p-value criterion. And so an agreement between the sponsor and our review division on the alpha is really the key, and it may be similar overall in the size of the trial that is required. From I think it is also important to remember regarding the discussion of real-world evidence that in addition to assisting with clinical decision-making, real-world evidence, if captured often in a randomized manner of administering the therapeutic, may assist with therapeutic evaluation for a therapeutic treatment effect. And the most important thing here, I think, is the reliability of the data sources and the variability that those data sources may introduce in whatever you're intending to study. And size clearly does not equal quality, but the pragmatic approach to clinical trials and real-world evidence may allow you to capture more events and more patients in a relatively more economical fashion. Clearly, we will continue to approve new agents for pulmonary arterial hypertension for 6-minute walk distance. We will continue to adhere to endpoints that address how patients feel, function, and survive. And this will include time to clinical worsening. And I will say that patient-reported outcomes, as has been mentioned, we will also continue to see as an approvable endpoint. However, as I think we heard in great detail, actigraphy although it has face value as a functional endpoint, it really requires a more work. It needs to be standardized. The monitoring output needs to be able to be comprehended, particularly given the diversity of devices, algorithms, analytic methodologies, and really the cornucopia of different outputs that are being developed and in this development space. And so there's really ongoing need for evaluation. I think that we as regulators look forward to better understanding risk scores as clinical trial endpoints and how change in risk scores really links to changes in clinical endpoints. And then finally, I want to reiterate what has been said multiple times here, which is continue to support novel development of endpoints. And I think that we are very interested in the idea of this disease modification endpoint and how it could be used to really address the proliferative pathophysiology of the disease. And we'd expect that a therapeutic that does permanently improve hemodynamics through disease modification would also have substantial benefits on clinical endpoints. And if these therapies do modify the disease process, then a randomized withdrawal trial, as we heard about earlier, may also be feasible. So I give back a little bit of extra time, but thank you so much for allowing me to speak here.
Okay, thank you. Thanks a lot for your nice presentation. And now we need to move to the last session, the patient viewpoint. And we have two speakers, Mrs. Caius Ovidiu Mersa from European Heart Network and Jacqueline Alikani from US. Each one 5-minute presentation.
Hello, good afternoon. This is Jacqueline Alikani from Los Angeles. I really have enjoyed all of the discussions today. Very intriguing, very informative, very educational, just amazing. And I'm a heart patient and I have a rare form of congenital heart disease. It's only been diagnosed in about 1 in 17 million people, and I was misdiagnosed for 48 years. And shortly after I was diagnosed with congenital heart disease, I participated in a research study funded by NIH. And in that study, it was a radiological study, and they diagnosed me with PAH. And then after that, I met several doctors who totally disagreed about the PAH diagnosis. So, as you can imagine, so this whole discussion today has been really interesting to me, and I learned a lot. And I also have a lot of questions. So I want to know particularly about the research trials that we discussed. I'm very curious to know more about the patient engagement on those research trials and the role that patients played in helping to design and develop the trials, assess the data, disseminate the trial results and everything. I'd like to know if patients and family members and caregivers were part of the leadership teams for the trials that were done. Because as Dr. Gibbs mentioned, as he discussed in his presentation about PROs, I agree that PROs directly from the patient are really, really critical. And it should really have a major primary role in all research efforts to improve care. And so I was especially struck by the patient-reported outcomes presentation in general, and the development of PROs and how the outcomes from PRO use outcome results are disseminated to improve clinical care. And I also think it's so critical to have the patients and the family members and the caregivers involved with research for a condition like PAH. I mean, when I was diagnosed, I had a lot of anxiety about it, and I still have a lot of questions because all of the doctors that I talked to, they don't agree and on the outcomes that were diagnosed for me. And so I went back to the research, one of the research team leaders, radiologist, and I reported back to him what the other doctor said, and he said he was right, he was sure, and he's very familiar with PAH and as a radiologist. And then after that, I went back to the other doctors and they still agreed to disagree. So I really appreciate this whole discussion, and because this is a life-threatening disease. And I joined a support group of PAH patients, and they have, share a lot of the concerns that I have about the status of research for PAH, and also better incorporation of patient-reported outcomes. And I think technology will help with that a lot, but the problem is with all the different technology and apps that exist, the standards development for that technology has not kept pace with the development of different apps and things for patients to collect their everyday symptoms and issues and report that back to their doctor during their regular doctor's visits. So again, I really appreciate this. I think it's a complex issue, and we have a lot of work to do in the future to help improve care for PAH patients.
Up next, the CVCT Forum multi-stakeholder moderated debate. What endpoints are clinically relevant, regulatory approvable, and economically valuable? Chaired by Valerie McLaughlin, Ann Arbor, USA, and Gérald Simonot, Paris, France.
One. Great. Well, thanks for a wonderful panel, everyone. Please feel free to raise your hand to ask a question. I'm going to start while you all think of your questions. And the question I have really has to do about the quality of life conversation. I think Simon gave a really wonderful discussion of some of the tools, some, FDA, developed with FDA guidance. The FDA is regulatory authorities are absolutely happy to look at that. Industry has used it not for primary endpoints. I just want to throw it out to all the stakeholders. Are we ready for quality of life as a primary endpoint? If so, we always have the conversation, what is statistically significant versus clinically significant? Where do you all think we are on that? Wendy, did you mean to respond to that question?
Yeah, I'm a firm believer in quality of life. We use it for our patients at every visit. And I'd be really keen if we could actually include quality of life questionnaires actually on part of the risk scores as well. I mean, it's directly linked to mortality. It's certainly linked to morbidity. And I think that quality of life, when you're talking to the patients, when you're having the shared decision-making conversations about going on to new therapies, quality of life is, as Simon said earlier on in his talk, it's the number one thing that patients actually want. And earlier on when Iona was talking about the holistic care of patients, not only are we looking at things like BNP and right ventricular function, but things like anxiety and depression, those also have a significant impact on patients' morbidity and mortality. So I think we really ought to be looking at their quality of life every time we see them. So I think it ought to be included in clinical trials, because if we're giving them a drug that improves their pulmonary vascular resistance, we also need to be making sure that it's not impacting negatively on their quality of life as well. So I strongly believe that we ought to be including it at least as a co-primary endpoint. But I think that we ought to be using it a lot more than we do.
Okay, Bjorn, did you have a question or a comment about that?
I actually have a comment to that. I think it's a similar situation that has been in heart failure. I mean, there have been drugs that have been improved patients' quality of life, but they shorten their life as well, and they have not been approved. I mean, I think that patient-related outcomes and the patient's quality of life is really important. And I think in heart failure, that has moved up the ladder very much in relation to mortality as a primary. But I think that to be able to approve the primary, the patient-reported outcome, quality of life, or that similar thing, you have to exclude in relation to regulatory that you don't shorten life. You don't have to show that you prolong life, but you have to show the safety that you don't shorten the life. I think
we've learned that lesson. Isabel?
Yeah, just to report what was said about quality of life. Basically, the payers in all countries define health as the combination of quality and quantity of life. So it is my very strong opinion, as the price of drugs increase, that both should be produced. If you want payers to accept the results of trials and to accept that they should pay something to get the drugs on the market. So it's really, I mean, increasingly we've heard from our authorities that they are not going to pay for a biomarker, they're not going to pay for cholesterol level unless it's really shown to be completely a surrogate for heart failure or heart attacks. So they want to pay for health. And health is defined by quality and quantity of life.
Well, I have a question on CardioMed for Ray Banza. Ray, for CardioMed, you measure only the pulmonary artery pressure without cardiac output, without systolic ejection fraction, or nothing, only mean PAP or systolic?
Mm-hmm. The three approved parameters are currently mean pulmonary artery pressure, PA systolic pressure, and PA diastolic pressure. However, we can glean stroke volume and cardiac output very accurately using the device. And those type of endpoints are in discussion with the FDA right now about approving for regular use with the device.
Okay, because if you measure only pulmonary artery pressure, I think it is not interesting in PH, because if you look at all the trial with hemodynamic, even with effective drug, you can see the decrease in mean PAP is only 3 or 4 millimeters of mercury. Increase in cardiac output and decrease a lot PVR. And it's not like for left heart disease, because when you have a variation in mean PAP in left heart disease is due to the variation of wedge pressure. And with wedge pressure increased by 10 millimeters mercury, the risk of pulmonary edema. In pulmonary arterial hypertension, when you are a decrease of mean PAP of 2 or 3, it can be due to an improvement of patient, but in many cases, it's due to deterioration, due to low cardiac output, decreased cardiac output. And in my mind, to have a technique measuring only mean PAP to follow the patient, for me, it's not applicable in PH.
Yeah, I agree with you, Gerald, and that is a nice thing about the contemporary algorithms with the CardioMEMS, in which you do get stroke volume and cardiac output. So you can follow pressures along with stroke volume and cardiac output. And one of those slides that I showed the beautiful correlation that you get with changes in pressure, improvements in stroke volume and cardiac output that are not related to heart rate changes. So you can really nicely show that using the CardioMEMS device now.
Okay, no, it's essential. I think it's very important to clarify, because many people understood is only the measure of mean PAP or systolic PAP. I think it's a big mistake. You need to be clear on this point.
Yes, yes, I tried to show that in the presentation, but you're absolutely right, Gerald. The incorporation of the stroke volume algorithm will be very, very important for PAH.
Thank you.
Yeah, that's absolutely a game changer, but I would like to throw that question back to Mitch, right? Because Gerald really nicely talked about how when we started, the FDA said no thank you to hemos, feels, function, survive. So Mitch, can you comment on the FDA's perspective on this device, please?
Sure. So I think that PVR and pulmonary pressures as they stand are not alone approvable endpoints. They are monitoring components of perhaps phase 2 markers of an effect, of a treatment effect, but will not really be feasible components of a clinical endpoint. I think that being said, the idea of this disease modification endpoint is where they could form part of an approvable endpoint, where if you can show a permanent change, even perhaps by withdrawal of the therapeutic in the disease process, that could be an approval endpoint in and of itself.
So Mitch, we have this discussion all the time, what is disease modifying? And you just said something that I've always thought is important to the FDA. You've got to have maintenance of effect with withdrawal of the drug. Is that the FDA definition of disease modification?
So I don't want to speak for all instances of what might be disease modification. I think that we are really always open to a thoughtful discussion with a sponsor on how they want to tend to or try to demonstrate disease modification. But I think that is certainly one way in which you could clearly demonstrate disease modification.
Ioana, it looks like you want to comment on that.
I know. I have to jump in because, if you look at rheumatology, disease-modifying drugs, they do modify the disease process, but they are never withdrawn, right? And they're approved. So I think we have to look at pulmonary hypertension because we do not have a cure. And even with the new therapeutic agents, I'm not sure we're close to a cure. But I think we may have a disease-modifying drugs. However, I'm not sure if it's safe to withdraw them. And I'm not sure if the definition of a disease-modifying drug is Once the treatment is implemented, you withdraw and the disease does not get backwards. So I'm not sure if that's the safest way to go, you know, and evaluate disease-modifying drugs in PAH by withdrawal with showing persistence.
Yeah,
Can I make one more
certainly have
comment?
angst. Yeah, please.
Can I make one more comment here? So I think that, as I said, I think there are multiple ways to do this. Another conceivable way you could do it is that a time-delayed transition from a placebo, for instance, if you delayed transition to the therapeutic, you could envision a therapeutic that may never be able to catch up with a previous treatment in terms of its disease modifying effect. So if you look at the time course of change, again, might be a way to demonstrate this effect. You know, the bottom line is that the defect is going to need to be demonstrated to know that it exists.
And then can I challenge you again that at any time in the disease process, you can modify the disease. That doesn't mean that you don't catch up. So if you start a little bit 3 weeks, 3 months earlier, may not mean that if you start 3 months later, you will not modify structurally the disease. So I'm not sure these two, withdrawal and catch up, are really, really significant, at least in the pulmonary vascular remodeling process.
I'll make one more comment in that it doesn't, you don't need to demonstrate disease modification to have a therapeutic approval. This would be for this indication. And so I think it's that negotiation on that topic, what quantifies disease modification, where we're having disagreement, but you don't necessarily need disease modification to have an approval.
Gerald, I know you're on a timeline. Do you have any other topics, any questions you want to bring up before you need to go?
I think that Fayez Zanad wants to interject right now before having another duty.
Yes. Thank you so much, Mark. I intend to just one observation. I have been watching your sessions, but commuting from one session to another, so I haven't seen all of it. But it's striking how much everything you have discussed today is being discussed among heart failure trialists, and I'm sure that Krish, Philip, and others who have been part of the heart failure trial as well can confirm this. And therefore, it is really my pleasure to have you guys at the CVCT meeting for this first time dealing with the pulmonary arterial hypertension. But the next step is certainly to have a common session overlap with the heart failure trialists, because it doesn't make sense that we are living in silos. Everything you have been discussed is already on the agenda and the table had to be discussed thoroughly with it, whether it is the choice of endpoints and what is relevant and the surrogate and all of this. And you have this exceptional, advantage of being still coined as a rare disease. Otherwise, it's exactly the same word than heart failure, same word than the type of trials. And this is a privilege that you have, because in heart failure, we have the tough objective in order to have morbidity and mortality trials every time we can come up with a medication. I'm sure that Krish can certainly confirm this. So I just wanted to thank you for the wonderful job you have done today, but encourage you to mingle with the heart failure colleagues, and maybe we'll make it happen next year. We have a common session between heart failure and pulmonary arterial hypertension trialists. Thank you in any case for the wonderful job you have done today.
Great. Well, thank you for having us. Let's move on. Hunter, you wanted to change the topic a little bit?
Yes, thank you, Val. So I just wanted to make a comment on activity monitoring, and thank Owana for nicely summarizing the current status of activity monitoring. And also, I think at the end, you came to the conclusion that you were not sure whether it was ready for prime time. And of course, I'm paraphrasing you there, but I think the same was said by Dr. Tsotsky. So, what I wanted to say was that, you referenced a lot of Balmer's work from Germany, and he and Thierry Troost are really, sort of key figures here. But Balmer published a paper which I think is really instructive. It was in IPF patients where they correlated some clinical measures with physical activity. And it was amazing how things like dyspnea, lung function, and so on were quite tightly correlated with physical activity. But when it comes to 6-minute walk, they grouped patients into those who walked more than 400, those that walked between 400 and 280 meters, and those that walked less than 280. And it was really interesting because if you walked more than 400, your steps per day were around 6,000 to 7,000. If you were in the next category down, 400 to 280, you also walked about 6,000 to 7,000. But if you walked less than 280, you went down to about 2,000 steps. And so I've always felt right now that activity monitoring is really for the more severe patients, and that's where we're going to see benefits with whatever intervention we choose. But I think we have to be really careful with patients who have pretty good walk distances, because physical activity, as you mentioned, Ioana, there are so many other aspects to it that I think it's going to be difficult to tease out differences.
Yes, I totally agree with you, Hunter. And not only, other issues, but temperature, less than 25 degrees Celsius, you guys have to translate into Fahrenheit, and humidity also make a significant impact on people's activity. The lower temperature and less humidity are associated with higher count steps during the day in the same person. And that was a very small study out of Berlin, which has, 4 seasons, right? But I would say, yes, the lower end and then the less active patients will always show improvement in whatever if you have an effective drug, whether it's walk test, 6-minute walk test, activity monitoring, hemodynamics, So those are the easy to show. But the higher end, if you attach an incentive program to improve their activity, I think that's where you're going to see the extra benefit of a drug in a lower risk patient, less severe patient, and less RV failure type of patient. So I think, and then we should study these patients, because one of the previous talks talk about how to identify earlier disease and treat these patients, because these were not going to be good candidates for clinical trials as we design them currently. But an activity monitor with an incentive to move more, because they will be able to move more, I think will be the way to test the less severe patients.
Great. Thanks for that. Jane Leopold, you have a question?
Yes, thanks, Val. I did. I mean, one quick comment, though, just to add on to Ioana's point, and that is that Evan Britton actually had a pretty nice study with an activity monitor and an incentive in PAH patients, showing that it made a difference in their activity and how much they move. So I would refer you to that for understanding some of the points about incentives. But I wanted to bring it back to the point that you raised initially, which had to do with quality of life. And I was particularly struck in Dr. Gibbs' talk about his comment that one of his patients said to him, "None of this applies to me." And so, and maybe get Jackie's input on this as well, but is it time that we start thinking about more precise or precision quality of life assessments and really finding out what the things are that the patients want to see improved and tracking those metrics over time?
So
Definitely, I agree with you because, you know, I don't see how so much research can be done without having, you know, the patients as equal parts of the research teams and giving feedback about their values and experiences and what they actually say they need to improve their daily quality of life, long-term and everything. I mean, this should have been happening years ago, and now we're in this whole new sort of patient-centered era, and I'm really excited and happy about it. But definitely, I think funders should mandate that as like PCORI does. If you don't have a patient engagement plan that is accountable and then you don't get funded. And there are resources out there like PCORI that exist that provide free services to assist the researchers.
So Jackie, it's Marty. I think you remember we were on the
okay, Marty. Yeah.
Yes, for a few years. I just wanted to step in because the SIMPACT study actually, had focus groups with patients and providers as to what was important. So it was really developed to be patient-centric. I don't think anyone is perfect. I think Camphor was clearly not reliable over time to show differences, but I do think that we as a community have tried to utilize these and have definitely put them into our new trials. And I do think that patients do need to be involved more, especially in phase 3 for the convenience aspects of things, because a lot of times we design trials and the feasibility of the patient actually taking the medicine the way that we've designed it is not always correct or good for them. And since I just stepped in, I do want to say that I think that Mitch Saka did a very nice job talking about disease modification, despite Awana's critique. But I do think that part of the issue is that those two ways of doing it is just, it frightens us. And I know that you take care of PH patients as well. And maybe we need to start moving towards disease modification of actually looking at the vasculature. And then we'd all be very happy to see that we've changed the vasculature and not just cause people to live longer.
So you think imaging would be the way to go, microscopic or, smaller, vision imaging?
Imaging or just even imaging on things that we're already imaging and doing a better job of it and letting the agencies utilize those images as part of our endpoint and disease modification. I think when we're looking for a new endpoint, we need to sort of step out of the box. And then finally, I still like my treadmill tests from 15 years ago because it's good for patients that are less sick and it's pretty easy. And then I'm going to stop talking because Awana, there are other people here that need to speak.
I want to go back. I want to ask Krishna and Mitch to actually comment on those quality of life issues, like, the differences we're talking about. Is there anything we can learn from other disease states that may have had a QOL as a primary endpoint of the study? I just want to know if there's something we can learn. I'm not sure if we have Krishna with us anymore, or Mitch.
He's there. I think he's trying to speak.
Yeah, I think his connection's not been good, is what they said.
Okay.
I'll speak up from the heart failure space, where quality of life, as we heard earlier, is also an ongoing discussion, and I think that the way it's been used there is a reasonable way. I think we would be happy to approve a therapeutic based solely on quality of life, as long as what's discussed earlier here, there is reasonable evidence that there is not harm on hard clinical endpoints. And I think you can look at the guidance document for heart failure endpoints for clinical evidence that was just published in the last couple of years from the FDA that goes through that discussion. And I think that we would be happy to include that. I think that what you see in the heart failure space, including recently, is a composite that includes mortality, hospitalizations, which are clearly patient-centered endpoints, as well as in a hierarchical series, patient-reported outcomes and quality of life. And I think that the way you analyze that is part of the issue, because the quality of life will inevitably be the driver of the overall composite. But I think we are very willing to look at that as an approvable endpoint.
The downside is I'm not sure how many patients we will be able to enroll to show a significant improvement in, in quality of life compared to the large heart failure outcome studies. But we should try.
Great. Thank you. Gerald, do you have any other questions or want to make any final comments? We're just about out of time.
It's late in France.
You're muted, Gerald. You're muted. You just need to unmute.
That's okay. I think we had an amazing discussion. Thank you, Val, for moderating.
Thank you,
and
Val.
Thanks.
In line.
Thanks. Gerald, I know. Okay.
Yeah,
Gerald, final words.
Thanks a lot, everybody. And we have to thank a lot Fayez Zanad for promote this meeting on pulmonary arterial hypertension. Thank you, Fayez, and see you soon.